Bismillahirrahmanirrahim. Assalamu alaikum. A very pleasant morning to one and all gathered here. Gaining knowledge is the first step to wisdom. Sharing it is the first step to humanity. With this inspiring thought, we cordially welcome the galaxy of intellectuals, the dignitaries, honorable guests, presenters and participants, respected staffs and students to the second international conference on the recent trends in biotechnology 2021, organized by the PG and Research Department of Biotechnology, 𝗕𝗏𝗄 College.
A grateful heart is the beginning of greatness. It is an expression of humility and a foundation development of such virtues as prayer, faith, courage, contentment, happiness, love, and well-being. Let us begin the occasion with the blessing of the Almighty. Now, I call upon 𝗕𝗏𝗁𝗲 of 3rd BSc Biotechnology, 𝗕𝗏𝗄 College, to recite a few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين صدق الله العظيم.
Thank you, Afra. We all feel so blessed.
Today is the day we begin to learn to look through the eyes of others, to find out and experience what the world is like for us. It is also the day we grow bigger than our differences and offer everyone a friendly welcome. As we begin today's program, I kindly call upon our coordinator, Dr. 𝗓𝗿𝗂𝗍𝗲 sir, to welcome the gathering.
Bismillahirrahmanirrahim. Honorable Secretary and Correspondent, Dr. 𝗕𝗶𝗁𝗂𝗍𝗲 Sahib, respectable Treasurer Haji M.T. Jamal Muhammad Sahib, and respected Assistant Secretary Dr. 𝗕𝗶𝗁𝗂𝗍𝗲 Sahib, beloved Dr. K.N. Abdul-Nihal, Member and Honorable Director, Dynamic Principal, Vice Principal, Additional Vice Principal, Directors of Hostel Administration, HODs, Professors, academicians, young researchers, and students friends. I am honored and delighted to welcome all of you to this second international conference on recent trends in biotechnology through virtual modes. Every year, Department of Biotechnology organizes conference for the benefit of students and research scholars with the theme of various development in the field of biotechnology. From last year, it is decided to conduct series of international conference with theme of recent trends in biotechnology. Today, we are in the second international of this series. Due to changing in time cause of COVID-19, the entire conference is planned to have on digital platform. We encourage our participants to stay safe and maintain social distancing. As a novel coronavirus continues to spread, scientists and researchers are working around the world to identify diagnostics, therapeutics, and vaccinations to help protect individuals and communities across the globe. Because of the sustained research development in the field of biotechnology, today, world can able to come up with a vaccine for COVID-19. World understands the importance of biotechnology field to the society. You work in biotech. Your work is more than just a job. You are doing something useful to the society, which is an incredible. This conference will be helpful to all young students, researchers, understand the challenges and opportunities available in the field of biotechnology. It's my pleasure to have today prominent speakers, Dr. 𝗕𝗶𝗁𝗂𝗍𝗲, researcher from Stanford University, California, USA. Dr. Nadesen Manickam, environmental biotechnologist from Indian Institute of Toxicological Research, Lucknow. Dr. Mukesh Thakur, scientist from Zoological Survey of India, Calcutta. Today, conference has about 240 participants participating from various institutions and around 25 research articles received, out of which 15 selected for today's presentation. I would like to congratulate the organizers of Department of Biotechnology for having organizing this today's second international conference on recent trends in biotechnology. I once again thank our college management and principal for the help in all possible means to conduct this today's second international conference successfully. Thank you very much.
Thank you, sir, for your thought-provoking address. Whenever you see a successful person, you only see the public glories, never the private sacrifices to reach them. Now, I call Dr. B. Naseema Banu ma'am, Assistant Professor of our department, to introduce our esteemed chief guest.
Good morning, one and all present here. It's a great honor for me to introduce our chief guest, Dr. 𝗕𝗶𝗁𝗍𝗲𝗍𝗲, who is our distinguished alumnus too. Dr. 𝗕𝗶𝗁𝗍𝗲 obtained his bachelor's and master's in zoology from 𝗕𝗏𝗰𝗲 College. He further specialized in human genetics and obtained an MPhil from Madras University. He went on to work at various capacities at the Center of DNA Fingerprinting and Diagnostic, Hyderabad, Institute of Human Genetics, and 𝗕𝗏𝗰𝗲 Institute, Cambridge. Dr. 𝗕𝗶𝗁𝗍𝗲 holds a PhD degree in molecular genetics from the University of Cambridge, UK. He carried out his doctoral and postdoctoral research under the supervision of Professor Fiona Watt, a renowned stem cell biologist at Cambridge and King's College, London. While in Watt lab, he conducted a first large-scale tissue-specific phenotype screen on hundreds of knockout mice and discovered many novel genes that are essential for mammalian scale function. He further elucidated the mechanistic role of sphingolipid and the ribosome rescue pathway in epidermal stem cell function. He has published many papers in the area of skin cell biology and won many awards, including a long-term fellow from the European Molecular Biology Organization and postdoctoral investigative grant from the Larry Hillblom Foundation. Dr. 𝗕𝗶𝗁𝗍𝗲 also holds a degree in philosophy, ethics, and theology awarded by King's College, London. It is perhaps these combinations of diverse background and training led Dr. 𝗕𝗶𝗁𝗍𝗲 to develop an interest in fundamental questions in neuroscience. He is currently an EMBO and Hillblom Fellow working under the mentorship of Professor 𝗕𝗏𝗁𝗍𝗲 at Stanford on genetic mechanism involved in synapse formation and function. He is also an avid communicator of science, eLife Community Ambassador, STEM Ambassador, and Open Science Advocate. He is a recipient of many honors and awards of international acclaim. Such a prominent person is among us today, and we are very proud to welcome you to your own alma mater. Thank you.
Thank you, ma'am. Felicitation is inspiration to others and motivation to whom you are felicitating. So to get motivated with great privilege, I invite Dr. 𝗕𝗏𝗁𝗍𝗲𝗍𝗲, Sir, EMBO and Hillblom Research Fellow, Department of Molecular and Cellular Physiology, Stanford University, California, USA, to deliver the inaugural address. Please, sir.
Can you hear me now?
Yes.
Can you hear me?
Yes, sir. Yeah. You can proceed. Yes, you are audible.
Okay, thank you. Thanks for the kind introduction, and it is indeed my privilege to inaugurate this second international conference on recent trends in biotechnology organized by the Postgraduate and Research Department of Biotechnology, 𝗕𝗏𝗰𝗲 College. Thanks to Dr. Bukhari and the other organizers for inviting me to open this conference and be one of the keynote speakers. Using this opportunity, I particularly extend my greetings to my beloved teachers and the management of 𝗕𝗏𝗰𝗲 College. The conference that we are attending today is designed and organized particularly for young students. I'm also pleased to see the focus that this conference places on supporting and encouraging the young students. I note from the conference program that themes ranging from environmental toxicology to genomics and proteomics would certainly offer the young students and researchers with valuable knowledge updates in the field. In this short inaugural address, I would like to cover some key aspects. First, I would like to begin with the history. Although the field of biotechnology is perceived as a modern branch of biology, indeed humans have used biotechnology since the dawn of civilization. So Egyptians used yeast to bake leavened bread, and Chinese developed fermentation techniques for brewing cheese and Aztecs used spirulina algae to make cakes. Today, when the most people think biotechnology, they probably think recombinant DNA. Although much of modern biotechnology does deal with manipulating DNA and RNA, classical biotechnology began long before even we know about genes and chromosomes. When began as a recipes for production of food, now include technology to enhance everything from farming to pharmaceuticals. That we think of a modern biotechnology began around end of 19th century. By then, Gregor Mendel's work on genetics was completed, and institutes for investigating fermentation along with other microbial processes had been founded by eminent scientists like Robert Koch, Louis Pasteur, and Joseph Lister. At the beginning of 20th century, industry and agriculture started to incorporate biotechnology. The growing automobile industry, for example, used fermentation process to produce acetone and paint solvents. By mid-century, major advances in genetics dominated biotechnology research. The discovery that DNA carries the genetic code and the structure of the double helix was discovered by Jim Watson and Francis Crick. Soon, new techniques were developed to allow manipulation of DNA. Genetically engineered plants, for example, microbes, animals, and products like insulin highlighted the future of this field. The importance of new genetics culminated in the Human Genomes Project. An international project, as most of you would aware, is instituted to map the human genome. The goal of this project is to identify structure of the entire human genome, including its 3 billion base pairs and approximately 22,000 genes. Similar to this, the International Knockout Mouse Project, arguably a massive initiative after Human Genome Project, offers to knock out every gene in the mouse genome so that the role of gene can be studied in a functional context. I have had an opportunity to contribute to this project in a small scale, about which I will be discussing in my research presentation in this conference. I hope that the knowledge will help the scientists identify, prevent, and treat many of the illnesses resulting from genetic malfunction. Today, biotechnology is being used in countless areas of research or fields, including agriculture, clinical applications, bioremediation, and forensics, where DNA sequencing is a common practice. Industry and medicines alike use techniques of PCR, immunoassay, and recombinant DNA. Genetic manipulation has been the primary reason that biology is now seen as the science of future, and biotechnology is one of the leading industries. And without doubt, students have unlimited career opportunities in these fields, be it research or product development. But in order to be competitive, students should mold themselves into accomplished candidates, which is obviously doesn't confine to classroom lessons, but comes up with even more responsibilities, which is my next message in this inaugural address, which is empowerment is the key. As you've seen from the history I outlined, there has been a lot of things happened in the past and a lot more going on currently in the field. In order to be successful, one must keep up with the trends and update the knowledge constantly. Students have enormous responsibility to constantly undergo learning process and empower them with the true knowledge. Students should be open-minded when it comes to learning. Today, they should be collaborating with other students, not just with the same department, but also other departments, so that they can learn from each other. I vividly recollect when I was a student of MSc Zoology in this very same college, I used to invite microbiology students to my room at the 𝗕𝗶𝗰𝗲 hostel to give seminar-like talks on microbiology topics that are not covered in my zoology curriculum. In return, I offered them to give seminars in ecology and entomology in their room in the new hostel. So this collaborative learning outside the classroom not only helped us to prepare and face many nationwide competitive exams, but also allowed us to think outside the box. No degree program is superior or inferior. One must utilize every single opportunity to learn from right source. When I talk about the source of information, I would like to reflect on the current scenario of information source. With an easy access to digital information, it is apparent that gigantic amount of information out there. All information is not knowledge. Students should also learn to pick the right information and weed out the wrong one so that it can become true useful knowledge and eventually a wisdom. One example is relevant to current COVID pandemic. There are a lot of misinformation out there regarding vaccines. Biotechnology students have responsibility in the society to clarify this misinformation more than ever. In order to do so, you should first know how virus infection works and how vaccines work, so that you are in a better position to explain it to your family and friends, so that your knowledge has a truly benefit to the society. This is just one example from one field of research. There are several examples to draw from. As Dr. Buhari rightly pointed out from his welcome speech, that you have the current situation and scenario of the COVID pandemic and the vaccine development is an example of how the students of biotechnology or biology in general can contribute to such a very necessary, timely necessary field need. So in this conference, I'm certain you will have an opportunity to learn from experts in wide range of research fields, right from environmental toxicology to genomics or proteomics. I would like to congratulate the organizers for putting together such an amazing program and talks, and I wish this conference a grand success. Thank you.
Thank you, sir, for your words of wisdom. Nothing is more honorable than a grateful heart. Giving is an expression of gratitude for our blessings. I kindly call upon Dr. 𝗕𝗶𝗿𝗄𝗎 ma'am, Assistant Professor of our department, to propose the vote of thanks.
Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Good morning to all. Today, by the grace of Almighty, I take this opportunity to put my gratitude into words. It is my great pleasure to thank our Honorable Vibrant Secretary and Correspondent, most respected Treasurer, Dynamic Assistant Secretary, most respected Principal, Vice Principal, Additional Vice Principal, former Principals, inspiring and energetic Dr. 𝗕𝗶𝗿𝗄𝗎𝗿 sir, member and honorable director of our college, directors of different functionaries for their constant support to organize this second international conference on recent trends in biotechnology. Thank you, sirs. On behalf of our college management, principal, and biotechnology department, I would like to extend my heartiest gratitude to our honorable chief guest, Dr. 𝗕𝗶𝗿𝗄𝗎𝗿 sir, MO and Hilblom Research Fellow, Department of Molecular and Cellular Physiology, Stanford University, California, USA, for joining with us in his busiest schedule. I add that Sir is an alumnus of our college. Thank you, sir, for enlightening us with your inspiring and motivating words. You are an exemplary person, and we have learned a lot from you, sir. Thank you, sir. I wholeheartedly thank our coordinator, Dr. 𝗕𝗶𝗿𝗲𝗿 sir, and member in charge of the department, Dr. 𝗕𝗶𝗿𝗲𝗿 sir, for their full-fledged support and guidance for organizing this seminar. Thank you, sirs, for assisting and encouraging all of us. Finally, my heartfelt thanks to the faculty members, research scholars, and students of our college and other colleges, teaching and non-teaching staff of our department for their constant support throughout this conference. Once again, I thank all of you and have a nice day.
Thank you, ma'am.
Now we are moving on to the technical session. It is indeed a great pleasure for me to invite Dr. 𝗕𝗶𝗿𝗿𝗲𝗿 to enlighten our young minds with a special lecture on mouse megagenetics, studying gene function at large scale. Sir, now the floor is all yours. Thank you.
Please, sir.
Can you see my screen?
Yes, sir. Yes, sir. Yes, screen as you are audible. You can proceed.
Okay, thank you. So thanks once again for the organizers to inviting me to give this talk. Today I would like to discuss about a work that I have carried out before I arrived in the USA, which was in the UK at the University of Cambridge and King's College London in the laboratory of Professor Fiona Watt. So the topic of today's presentation is mouse megagenetics, and why I call it as megagenetics, you will be able to appreciate from the talk that I'm going to, from the results and the methods that we use, you'd be able to appreciate that. And how this is helping us to understand gene function, not just one gene at a time, but at the same time we can understand the many gene function at a given time. So in this presentation, I'm going to talk through some of the main aspects of this presentation. So first, I would like to briefly introduce the audience to the knockout mouse project and mammalian skin, because my expertise that from my previous position is looking at mammalian tissue function with an example or as a model system of mammalian skin. So throughout this talk, I'll be giving an example of how the knockout mouse project helped us to understand mammalian skin in a molecular genetic context. Next, I will introduce to you the method, which is a genome-wide reverse genetic approach, how it helped us to identify novel skin phenotypes in relation to the specific gene knockouts. And I will also elaborate about a specific gene or a molecular pathway, how that helped us to really identify an evolutionarily conserved ribosome rescue mechanism in mouse epidermal homeostasis. So most of you are biotechnology well-learned students will know what are the similarities in terms of the genomic similarities between human and mouse. So we know mouse genome shares remarkable similarity with that of human, so that makes the laboratory mouse suitable to study human gene function as a suitable animal model. So mouse gene eventually could help decipher human diseases. To briefly introduce you to the mouse genome gene knockout technology, I like to draw your attention here. I'm not really showing an extensive technical detail, but briefly, if you look at this in the left panel, you have a wild type mouse where you have gene of interest in 2 alleles that would lead to a functional expression of a protein sequence. You could imagine any gene of your interest or any protein of interest should follow this molecular logic. So in order to understand a function of a gene, one way to do is actually to knock out, meaning you could delete or manipulate the functional or the protein output of the gene. So in this case, on the right side, you see a knockout mouse where the gene of interest has been deleted or knocked out in 2 alleles of the chromosome. Now, the functional expression would result in the protein with 1 amino acid substitution, or one amino acid substitution is shown as an example here, but it could result in complete ablation of protein expression, meaning that gene is not expressed. So with this in mind, a large-scale mouse genome mouse knockout project aims to create resources for large-scale gene studies. So this is a schematic of how those resources can be used to understand the biology in general. So from the left panel, you see how new mouse mutants are generated by microinjecting the gene knockout construct into the embryos. And then you have these many mouse mutants. In this case, it's 900, but the mouse genome mouse knockout project has reached more than 2,000 gene knockouts to date. Once you have those knockouts, what you could do is you could subject those individual knockout mice to various levels of phenotypic screen. To begin with, the top is an example of a metabolism. If you knock out a mouse, how that would lead a mouse to gain obesity, so that you're actually studying the obesity in absence of that gene, so that it conveys a message that that gene is implicated in obesity. And the same goes with the musculoskeletal system, sensory, and the defects in the blood cells, development, and even in the expression. So when these resources generated, the mouse resources are generated, these resources are made open access, which means any researcher from anywhere in the world have access to these mice, so that they can analyze their gene of interest or their knockout of interest in much more detail, depending on what they are interested. So the eventual output of this would be a new biological knowledge. And also, as we know, mouse and human genomes are overlap in a high similarity. One could always associate human diseases with these knockout mice diseases, which will also you will see an example from my presentation. So with that, actually, I wanted to jump into my model system, which is a skin. So this extraordinary painting in 15th century, the artist has depicted the importance of skin. Skin is something that we take it for advantage because we don't appreciate how important organ it is. It is one of the largest organ in the body, in our body. So if you have any defect in the skin, it eventually affects the other system of the body. For example, skin is an integral part of the vascular system, the immune system, or musculoskeletal system. So altogether, this exemplifies this art exemplifies how important and how massive the skin tissue system is. And obviously, I'm not jumping into many diseases that are related to several skin conditions, but I would move on to the introduction because in order to understand my presentation, it is important that you have an awareness of how the mammalian skin looks like and what are the components. So in this schematic, it has shown a rough diagram of the cross section of mouse skin. As you see, mouse skin is made up of two major layers. One is the epidermis and the dermis. The epidermis is the outer layer that you can see from your eye. That's the outer layer of the skin. So the epidermis is also home to hair and hair follicle, as you see here. So some of the stem cell populations that are marked in different colors are just examples of few stem cell populations, but there are more stem cell populations reside in hair follicle and in the epidermis. So when the dead cells are shed out from the outer layer of the epidermis, the stem cells resides in the basal layer of the epidermis are responsible to give rise to a new cell that moves upwards migration and then divides through the mitotic process called this whole process called differentiation. So the cells in the bottom layer are stem cells that not only give rise to committed cells to differentiate, but also they self-renew themselves so that the stem cell population number is maintained. Now, the hair follicle is another important organ, a mini organ, given that the number of various stem cell population. So here, the LGR6, LRIG1, PLET, these are the markers that are specific for the stem cells in these hair follicles because they're heterogeneous nature of various stem cells in the hair follicles. So in the stem cell field, the hair follicle is known as stem cell zoo, which means it has a range of stem cells that are responsible for maintaining the function of the hair follicle and the skin in general. So in this confocal image, 3D image of a mouse hair follicle, what you see is the full structure of hair follicle with infundibulum that connects to the epidermis and a junctional zone between the sebaceous gland and the bulge region. The sebaceous gland is nothing but a sac of oil that is responsible for oiliness of the skin. So if something goes wrong in those cells that you see that produces the sebum-like oil rich substance, you'll have bad skin. Same with the bulge region of the hair follicle that is home for various stem cell population that gives rise to new hair every time a hair falls out. That's called hair regeneration. So this organ makes a very unique and easy system compared to other systems of mammalian organism, allows this to be a favorite model system in adult stem cell biology field. So we use this as a model system to address the range of knockout gene function in our future talk. So why studying unknown gene of skin is important? So that's because the role of large number of genes remain unknown in mammalian skin function, and more than 100 human genetic diseases affecting over 20% of the population are characterized by abnormalities in the skin. So current genomic bandwagon effect may not be useful to catalog the function of unknown genes, which means a bandwagon effect means that if one gene study is carried out in one laboratory, everyone wants to study the same gene in their laboratory. So that's called genomic bandwagon effect. So that the genes that are not studied have been, overlooked and ignored. That's called ignorome. So completely uncharacteristic set of genes, the ignorome requires an attention to find how their expression and functions differ from well-studied genes and pathways. So adapting an unbiased screen to uncover the function of ignorome genes. So my first part of this presentation would be going through the genome-wide reverse genetic screen that I have conducted as part of my research in Watt Laboratory to identify novel skin phenotypes. So the pathway, the pipeline and the workflow looks like this, the strategy. So when the knockout mice are made, so this was with a close collaboration with the Wellcome Trust Sanger Institute, where I used to work before making these knockout mice transgenic vectors. So once I moved to Professor Watt's lab in Cambridge, we had an opportunity to use these mice for my study in the laboratory, where we used the mouse tail as a again a model system within skin to address the question, the genetic question that we had in mind. So what we did actually, we did a comprehensive tail skin phenotyping, where we looked for keratin 14 expression, which you have seen from my previous image, which is uniformly expressed in the epidermis, epidermal basal layer where the stem cell are. The keratin 14 expression again specifies the unique bulge region where all the hair follicle specific stem cells reside. And we also looked at hair cycle, hair follicle pattern, sebaceous gland, which I just explained, and the interfollicular epidermis, which is a region of epidermis between two hair follicles. So once this data is gathered, this has been fed into a centralized mouse resource portal, where any researchers can access the data, and then not only the data, they can also have access to the tissues that we have generated from the screen. So initially, the knockout mouse project started as a Sanger mouse project, and then it became International Mouse Phenotype Consortium, which any researcher can from anywhere in the world can access to the data generated. Researchers like me who have deposited data there could be able to get under their gene of interest. Now, how the pipeline looked like. So when we received the mouse tail skin, what we did, we separated that into three portions. First portion would go into the 24-well format rapid epidermal immunostaining, where you could stain the epidermis with the keratin 14 and keratin 15 markers that would give you the beautiful architecture of the epidermis, including the hair follicle, with a confocal microscope image. And then the second half has been the tail skin has been stored for deep paraffin histological sections, and then finally the genomic DNA samples can also be obtained from the tail samples in case if we need to confirm the genotype through sequencing and the PCR analysis. So this is an example of how we looked at the hair follicle and the hair bulge region that would have an immense functional relevance to the epidermis and the skin whole, where you see here, as I mentioned, keratin 14 that marks the epidermal basal layer, where basically you're looking the epidermis from the bottom, where you see the unique expression of the keratin 14 in red throughout the basal layer, where you have stem cell population. And the keratin 15 marks the hair follicle bulge, as I mentioned, the stem cell reservoir of the hair follicles. So this will give you the the overall pattern on distribution of how skin and hair epidermis and hair follicle pattern is organized, because many skin diseases arise due to abnormal hair follicle pattern formation. So from this screen, what I have obtained is, after analyzing 538 knockout mice one by one with this imaging method, we could be able to identify several novel skin phenotype. So for example, on the top here, you see there are different genes, and the red mark denotes the abnormality obtained in these categories. So for example, here in the tail epidermal hormone, what we have seen, this, for example, this 𝗕𝗶𝘁𝗲 gene has abnormality, but it doesn't have an abnormality in the other categories. Whereas the 𝗕𝗶𝘁𝗲 gene has abnormality in skin dysmorphology and tail epidermal hormone. So this is actually an example of how, after analyzing the knockouts of all these 500 knockouts, we were able to catalog their abnormality in different categories. So one of the examples here, diverse phenotypes identified within epidermis. For example, as I mentioned, abnormal hair cycle in this yet to be named gene with a number has rather different hair follicle size compared to the wildlife controls. Whereas here, another example called RAD18 knockout, where you have, instead of triplet hair follicles in the tail, you have only one hair follicle, which is a very interesting developmental phenotype. Another example is looking at this JHDM1, how the hair follicle morphology is altered, tells that this gene may have a role in the hair follicle development. And there are multiple other examples, and which I don't want to go through, but I hope you would appreciate how this gene knockout method helps to understand to different or many genes in a short period of time. So this is the almost final conclusion or observation from my study. So how many genes that we analyze, how many knockouts, and how many of them correlated with human skin disorders? So for example, we've identified mutants associated with human disorders as 140 of 538 genes, of which we had mutants associated with human skin and hair abnormalities were 45. So the actual mouse skin phenotypes that we revealed, meaning that of 538 gene knockouts, we found 50 genes that are responsible for, one way or the other, maintaining skin function in general. Interestingly, mouse skin phenotypes overlap with human skin and hair abnormalities were 9. So this is a remarkable number, which means you have identified 9 genes that have a disease relevance in human through this mouse phenotype screen. So some of the genes are here listed, pretty much all of the genes. So for example, some of the gene mutations in human, 𝗕𝗶𝘁𝗲, which has abnormal pallor phenotype, which is related to a skin condition. For example, 𝗕𝗶𝘁𝗲 mutation in human have a sparse or absent hair. So these are the human phenotype ontology specific terminologies that allows us to map the skin phenotype that we observed from mouse. Now, what are the molecular relevance? Okay, we identified skin phenotypes from the gene knockouts. So what is the molecular relevance? So when we looked at the genes with phenotypes, the 50 genes with the most significant phenotype, we subjected those 50 genes into signaling pathway analysis, where we found that many genes were mapping for the epidermal growth factor receptor signaling pathway, and some of them were notch and wind. So what this tells is these signaling pathways are essential for developing mouse skin or skin stem cells to maintain their function. So next, I wanted to quickly go through few examples where we identified truly relevance to human skin condition. For example, if you take in myosin 5A mutant, where myosin 5A has been knocked out, on the left you see a wild type mouse where it is black, whereas the mutant mouse are completely different. They are more of like a silvery gray condition. So this is a very interesting phenotype. Okay, what it tells, it not only just alters the coat color of the skin, but it also alters the coat color alteration comes from the melanocytes resides in the epidermis. So if you look at it in the wild type here in the bottom panel, where you see the confocal image of the epidermal hole mount, where you see these dark spots that they are not visible in the wild type sample, but in the myosin 5A knockouts, it is clearly apparent. So they are nothing but melanocytes. They are readily visible. So it doesn't mean that the wild type mice doesn't have a melanocyte. They do have a melanocyte, but they have a reasonable amount of melanin deposited in those melanocytes or produced in the melanocyte. Whereas in the myosin 5A, you have an abnormal deposition of melanin pigments in the melanocytes. So that is readily visible by this microscopic image, both in this panel and in this bright field where you can see a dark pigmentation. Okay, so what myosin tells about this? So myosin, as most of you would know, is a motor molecule. So in the pigmentation process, it is known that when the melanin pigments are synthesized in the melanocytes, they need to be transported away from the melanocytes to the neighboring keratinocytes, which makes the epidermis, in order to have a uniform skin color. So what happens is the myosin as a cargo molecule carries these melanin pigment or melanosomes to release from the melanocytes through the exocytosis process, so that melanosome particles can be uptaken by the neighboring keratinocyte, so that there is a uniform deposition throughout the skin. So in the absence of myosin 5A, it would be apparent that you don't have a cargo transporter to carry out these melanosome transportation. So you have the phenotype. Okay, now what is this? How this relates to human disease? So in 1999, a disease was identified in human babies that's called 𝗜𝗏𝗁 or silvery hair syndrome. So the silvery hair syndrome hair is not a trait of neither of the parents, so that it is unlikely the baby should be inheriting from the parents. So they have this baby born with this silvery hair, and they found actually this syndrome or disease is caused by a mutation in myosin 5A. So this makes our study hugely relevant to human genetics to address any therapeutic possibilities to treat this condition. Now, next I have multiple other examples, but in the interest of time, I'm skipping. But we also identified another gene through this phenotype screen, which is myosin 10, which is also involved in the mouse pigmentation. Another different category of gene is called alkaline ceramidase, which we found is essential for mammalian skin homeostasis and importantly regulating the whole body energy expenditure. This is an interesting study where we have found if you knock out this alkaline ceramidase 1 gene in those mouse, they not only have a defective skin that you can see by eye, but they, if you subject those mice into high fat diet, their wild type counterparts, control mice, would gain obese like body weight, but these mice will not gain any weight at all. So that intrigued us how a skin defect could lead to mice not gaining any weight, although when it eats a high fat diet. So this study would help us to understand, if you have a defective skin, you have an elevated level of water loss. So when the water is lost, the body temperature, the mouse struggles to maintain the body temperature. In order to maintain the body temperature, the mouse burns the fat that it eats from. For example, the fat is deposited in the form of white and brown adipose tissue. So in order to keep the body temperature, the mouse burns its fat so that it doesn't attain any heavy body weight. So this is a very good example when one system affects, one gene affects one system, how that influences other system, in this case, a metabolic system. So another example where we have shown, or we have used the resources generated from this phenotype screen that helped us to study the 𝗤𝗂6 positive cells. 𝗤𝗂6 is a transcription factor specifically expressed by some subpopulation of cells, how that helps to acquire stem cell properties in the epidermis. So to summarize, I have, from this part of my presentation, I have showed how a first large-scale mouse phenotype screen on a specific tissue is performed. The second, gene discovery in epidermal differentiation, for example, keratin 76, which I haven't discussed in detail, but that's an example to give you away. And the discovering genes that have a prominent role in pigment function, for example, myosin 5A and myosin 10, mapping skin phenotypes to human diseases, and vice versa, meaning that human disease to skin phenotypes. We found 9 skin disease models with mouse phenotype. We also found environmental influence of skin phenotype, how a diet can affect the mouse, and we used this TM9SF4 gene knockout as a model to study that. The potential interaction of mutant genes with EGFR and Notch and WID pathways. So the power of phenogenomics, that is combining the phenotype studies with the large-scale genomics in understanding unknown genes involved in tissue function, is exemplified from my study. So I would like to next quickly jump into completely different genetic study, but again, it is linked with the phenotype study that we have performed. So in this talk, I'll be completely focused on an ultramolecular pathway. I'll be not talking about any phenotype screen or anything, but this will delve into very interesting discovery that we made by using the knockouts that have been generated from the International Knockout Mouse Consortium, which is a role of an evolutionarily conserved ribosome rescue pathway in the epidermal homeostasis. Okay, so let's switch the gear and look back to recollect our molecular biology lessons that you might have learned from your classes. So we know the protein synthesis is a highly very sophisticated and highly controlled and high fidelity process, which means each and every step of protein synthesis, from initiation, elongation, termination, all these steps are highly controlled. If something goes wrong in any of these single step, you will not have a proper protein production. Okay, so in this case, for example, you have a normal translation cycle. You have the ribosome complex comes in contact to each other. In this case, the 40S and 60X complex. And then you have a start codon, and then you have a smooth translation. And then you have a polypeptide produced or synthesized or translated from mRNA. And then after the end of the translational cycle, you have a dissociation of these two complexes, so that these same ribosome subunits will be recycled for next round of translation. So this is how a normal translation should occur in the cell. But what can happen is some events can cause the ribosomes to stuck in the mRNA. Okay, so in this case, a truncation, because it doesn't have a stop codon or poly A tail, or an obstruction or damage in the middle of this coding sequence where you have a secondary RNA structure. So the ribosome is stuck, it doesn't know what to do. Or a tRNA insufficiency. We know the tRNA, amino-acyl tRNA are essential to decode the mRNA into amino acid. Or a poly A ORF in the poly A in the ORF. So poly A should be after the stop codon, but if you have an unexpected poly A in the open reading frame before the stop codon, the ribosome will not move further. So this whole overall process is called ribosome stalling. So what can happen if something happens in the cell? So it can trigger pathways for mRNA degradation, ribosome recycling, protein degradation, stress response, or ribosomal degradation. So one of the complex that is involved when ribosomes are stalled unexpectedly in the on the mRNA. So there's a protein complex called HPS1 and DOM34. They come and rescue the stalled ribosome so that the abnormal protein production is not carried out, or the ribosomes becomes available for the next round of translation. So DOM34 so this whole mechanism has been well explored in yeast. So HPS1 and DOM34 are yeast discovered proteins, but DOM34 is known as pelota in mammalian system, both in human and mouse. So it rescues this complex rescues stalled ribosomes for recycling. So in yeast, it's been known that in the absence in the event of ribosome stalling with a truncated mRNA, these DOM34 and HPS1 complex comes and rescue the ribosome, so that ribosome becomes available for the next round of translation. Now, in the DOM34 knockout mutant in the yeast, truncated mRNA with the stalled ribosome undergo a failed ribosome rescue and recycling. So a readout of this ribosome stalling can easily be seen or easily be analyzed as a short mRNA footprints. So what happens if you have a failed to rescue the ribosome? Downstream pathways is largely unknown. So they didn't know in the yeast if this leads to any protein synthesis downregulation or translational downregulation. On the other hand, in the mammalian system, what is known so far from some studies from labs around the world, so they have also shown a similar mechanism exists in the mammalian system, where you have the pelota instead of, DOM34. It's an analog of or ortholog of the same gene, where the mutant is the function of the pelota mutant in mammalian system is not known. So it has been known in some studies that the pelota is involved in embryogenesis and adult stem cell function. For example, in this study from Ibrahim Adam's lab in Göttingen, Germany, that they have shown that this gene deletion causes early embryonic lethality and defects in cell cycle progression. And subsequent studies from their lab have shown that it has a defect in the spermatogonial stem cells. And by collaborating with our lab, because we had an expertise in analyzing the various knockout mice, so they have found that with us, they found that this pelota regulates the epidermal differentiation. So we wanted to really look at much deeper how this pelota regulates epidermal stem cell function. So we utilized yes, the pelota is ubiquitously expressed in all skin compartments. So to utilize the genetic knockout technology to ask the question about pelota so that we can ultimately understand the ribosome rescue mechanism, we deleted pelota in the epidermal specific way, because full deletion of pelota, as you see from the previous slide, causes embryonic lethality, which means it doesn't allow this embryonic progression. So the knockout mouse technologies allows you to delete the same gene later in the developmental time point. So in this case, or the same development, but it's called conditional knockout mouse strategy, where you can delete the gene specifically in one tissue compartment. So in this case, the pelota knockout on the left side mouse, which is a conditional because it's still not a complete knockout because the pelota is not deleted. Once you cross that mice with a 𝗓 expressing mice under the promoter of keratin 14, as you have learned from my previous results, that keratin 14 is specifically expressed in the stem cell compartment. So now what happens is the 𝗓 is, when these two mice are bred together, the 𝗓 is expressed through keratin 14 promoter, will recombine these two red triangle here called the 𝗥𝗶𝗁 sites that will allow excision or deletion of this pelota gene from that particular cell, which means all keratin 14 expressing cells are absence of pelota. Now you can really study that function, how that affects the epidermal function. To our surprise, we had a dramatic phenotype. So in this case, this is postnatal 40 days, mean 40 days old mouse. You have a control mouse, whereas you have pelota specifically knocked out in the epidermal compartment through the K14 𝗓 mice, have a severe skin condition with skin hyperplasia. So as I mentioned, that one functional assay that one could do to assess the quality of the epidermis or quality of the skin is through the transepidermal water loss assay. It is nothing but a probe that you have, a kind of a small handheld instrument you put on the skin, and then it will assess the amount of water loss is happening in that given time. So as you see here in the control mice, the level of transepidermal water loss is much lower compared to the pelota epidermal knockout, which tells that this mice doesn't have a proper epidermis. And obviously, if you look at the anatomical sections of these mice, you see a significant thickening of the epidermis, which means the epidermal hyperproliferation, meaning that the stem cells don't undergo proper proliferation. Instead, they produce more unfunctional cells. Now, what happens if you delete pelota in the other skin compartment, which is dermis, which is the lower layer of the epidermis, lower layer of the skin, where it has a completely different type of cells. They are fibroblasts, not keratinocytes. So now we ask that question, okay, what happens if we specifically delete pelota in the dermis by crossing those mice with the dermo1 𝗓 mice? To our surprise, we did not see any major difference in the dermal compartment. So this tells that this is specific for the epidermal function. We further went on to confirm this phenotype in human epidermal stem cells, where we knocked down artificially human pelota in the cells cultured from human skin samples, where you see abnormal growth of those stem cells in the dish compared to the scrambled control. So what you see here is a kind of a fake human skin, meaning that only the dermal part is a proper obtained from a proper skin. Then what happens in the laboratory is you can scrape the epidermis completely off from the dermis, and you use the dermis as a scaffold to grow these artificially cultured cells that would eventually look like a skin or human skin. As you see here, the keratinocytes in the pelota, say RNA control, have a hyperproliferation, and compared to the controls. So this tells that this not only unique or not only specific for mouse skin, but it's also specific for human skin. And here the proliferation of those cells has been marked with several markers, for example, 𝗕𝗂𝗁𝗲, 𝗕63, and other markers. Now, we went on to really probe deep into this molecular function of this pelota. What happens if you transiently delete, meaning that you're not deleting pelota in the skin from birth. Instead, you're deleting pelota at this later time point. So what you can do is, instead of using a 𝗓 mouse, you can use a 𝗓 because the 𝗓 is fused with estrogen receptor, which means if you add 4-OHT into the mouse skin, it will transport the 4-OHT is nothing but a tamoxifen, which binds to the estrogen receptor and take this 𝗓 into the nucleus so that it will recombine the pelota locus so that pelota will be deleted. Excuse me. So this gives a very good opportunity for us to delete pelota whenever we want. So that's what we did. So we further showed after the mouse is developed completely, you shave the skin and then you apply tamoxifen or 4-OHT here, and then you observe the abnormality developing from the tamoxifen treated area. So then again, you can use the transepidermal water loss assay to measure the water loss from the treated area where you have a significant increase, which tells that epidermis does undergo abnormal function. Now, in this data, we also extensively characterize the sebaceous gland abnormality from this K14P, which I have previously introduced why that is interesting and why it is important. And I also show here with the various sebaceous gland marker, for example, fatty acid synthetase N gene expression, which is high in the control mice compared to the pelota epidermal knockout mice, where it loses its expression or differentiation capacity. Same goes with the other stem cell and the differentiation marker, where you have a less expression. Now, we further went on to really look at how the proliferation itself is affected. So one way to study the proliferation is actually you can inject the mice with EDU, which is a ethyl uridine analog, which binds to the uracil of this DNA base, where you have constant labeling when the cell divides, meaning that you can, anytime after the injection of this period of time, you can look for the proliferation of those population. So in this case, that's what you see. So in the wild type, you see not only abnormal hair follicle, but you also see abnormal cell proliferation. So conclusion one from this actually, that pelota phenotype is highly specific for the epidermis, and it is not for due to any systemic defects. So what is the role of these different stem cell populations in pelota epidermal phenotype? So as I previously mentioned, that hair follicle and the mammalian epidermis is controlled by many stem cell markers, stem cell populations. But I'm giving you here three well-known, well-characterized stem cell populations, which is important to follow my further results that I will show in a bit. So LGR5 is unique to the lower region of the bulge, and LGR6 is in the middle, and it's also present in the interfollicular epidermis, and LRIG1 populations are unique to the junctional zone of the hair follicle and the sebaceous gland. So just to imagine the potential of this population, meaning that if you isolate this LRIG1 population from the hair follicle, they have the capacity to produce entire new epidermis and the hair follicle. So that's why these stem cell populations are important. So we asked actually, what happens if we delete pelota in these three populations separately? So to our surprise, what we found, 𝗏 deletion in Eldridge specific population led to a dramatic, significant abnormality compared to LGR5 and LGR6 population. So this is really interesting. So 𝗏 is not only specific for epidermis, but within the epidermis, it has a differential functional role in different stem cell compartments. So this is what it tells. So again, this has been shown with multiple markers that are known to label the proliferating cell populations, and we find that 𝗕67, 𝗕63 populations are high in the 𝗏 deleted in Eldridge compartment or Eldridge population. Now, when we again use the transepidermal water loss as a functional readout to assay the functionality of the skin, you can see Eldridge 1 population has the 𝗏 deleted in Eldridge 1 population mice have a significant increase in the transepidermal water loss. Now, I'm not going into deep into this data, but it tells that how these different transgenic technologies or different transgenic mice, in this case Eldridge 1, LGR5, and LGR6, can be used to study the 𝗏 abnormality in the skin population. So conclusion 2 from this presentation is, we know that it selectively, the deletion of 𝗏 selectively affects subpopulation of epidermal stem cells, but we always interested in the molecular mechanism, like why, because as I introduced to 𝗏, why and how this affects the actual translational machinery. So in order to address that, because it is related to ribosome biology, so we collaborated with a lab in Johns Hopkins University in Baltimore, USA, where they have extensively studied DOM34 role in yeast, where they have used the method called ribosome profiling, which means you can really analyze the ribosome occupied mRNA fragments, how they are, the sequence of it, how big, how small, all aspect of it. So when we collaborated for 𝗏 knockout project, what we found that the ribosomal occupancy of the mRNA, it's called ribosomal footprints or RPFs, reflects a failure to clear stalled ribosome and failure in ribosome recycling. So the short ribosome footprint indicates that in the mammalian cells, in absence of 𝗏, like DOM34 in yeast, it tells us that the ribosome stalling, ribosome rescue upon ribosome stalling is failed. And obviously, what are those mRNA? What are those mRNA codes for? And then we found several of those are related to genes that are related to skin, for example, collagens, histones, and keratins, and so on. Finally, we found that while affecting the ribosome rescue, this 𝗏 absence in those population of cells led to a difference in the protein translation. As I alluded before in the introduction, how this specifically affects the translational machinery of protein synthesis. What we found is actually the protein translational efficiency is increased when 𝗏 is knocked out. And also we found several, the signaling pathways that are related to protein synthesis. Now, how to visualize protein synthesis in the tissue? So what we found from the previous results is through the RNA sequencing with the ribosome occupancy, that we found some of the transcripts are upregulated that are related to protein synthesis. But in order to see the protein synthesis by eye in the visual under the microscope, what I did actually, I have injected the mice with the OP-Puro. So OP-Puro is nothing but the puromycin analog that binds to actively translating or actively synthesizing amino acid or polypeptides, so that through some chemical conjugation methods, you can label those newly synthesized proteins, and you can clearly see how 𝗕𝗏 deletion leads to increased protein synthesis in the epidermis and in the hair follicles as labeled with OP-puro. So here with the flow cytometry, after isolating these cells, what we have shown that it's indeed integrin alpha 6 high population, which is a stem cell, another stem cell marker that is been widely used in flow cytometry experiments, shows that 𝗕𝗏 has a high amount of protein synthesis rate. So deletion of 𝗕𝗏, again, it also leads abnormal cell morphology, where you have an example with the epidermal sheet from the ear skin of the mice, where we show that it leads to a larger cell size. So the conclusion is, the final conclusion is, the 𝗕𝗏 phenotype is due to altered global protein synthesis, global transcription, and also protein synthesis levels. So can we reverse the phenotype by controlling the protein synthesis level, which comes to final part of my presentation. So protein synthesis is well known to be controlled by the mTOR pathway, the mTORC1 pathway. So you can easily inhibit this pathway, meaning if you inhibit the expression of mTOR complex 1, you would affect all the pathways or components that are essential for the protein synthesis. So one of the drugs that inhibits mTOR complex is rapamycin. So when you inject rapamycin into these 𝗕𝗏 knockout mice, you don't reverse this phenotype of the abnormal hair follicles, but you do reverse the epidermal proliferation when it is treated with the 𝗕𝗏, when it is treated on the 𝗕𝗏 knockout mice. So this tells that the rapamycin treatment can successfully stop the progression of the abnormal phenotype. Now, and again, this has been produced with the different other, the K14-CrR mice I have explained, where we have seen a significant reduction in rapamycin treated mice compared to the vehicle, which is a control or a mock treatment where you don't have a drug. Whereas you have rapamycin treatment, treated mice, you have almost half of the thickness of the skin, which means that somehow the inhibition of mTOR pathway reduces the proliferation. Thus, it gives a partial rescue of the phenotype. Now, this has been shown with several other markers of proliferation and the mTOR. So we know that. So I would skip this aspect of the project because it has, although it has a relevance to what I have spoken, but due to the interest of time, I'm going to skip this. So to summarize what I have spoken on the role of 𝗕𝗏 in the, not only in the molecular context, but how you study the molecular pathways that you study in textbooks have a profound implication or profound importance on the overall tissue function. So that's what I have shown here with the 𝗕𝗏 as a model system, how the deletion of which leads to failed ribosome rescue and recycling, and short mRNA footprints, and pervasive or global transcriptional changes with a global increase in the translational upregulation. Now, this is pretty much what I have explained in the summary. I would like to end this presentation by acknowledging my laboratory where I have carried out this research in the University of Cambridge and King's College London, especially Professor Fiona Watt, who is here. This picture has been taken several years ago during our lab retreats where I was a PhD student. And as you see here, the project itself is a highly collaborative project. We have collaborated with multiple different international research institutes with ambitious research programs. So as you would be able to appreciate from my talk, that science is highly collaborative and it's truly international. So every country have its own expertise and the resource which can be used if it is used in a correct way. And also what you would have appreciated from my presentation is that several techniques that are recent, some of them are recent trends. For example, the ribosome profiling didn't exist, the method didn't exist until late like 2010 or 10 years ago. So it is a very recent and it is constantly evolving, constantly improving. So these are the trends or methods that are new. And I also covered the RNA sequencing. So most of you would be aware of DNA sequencing, but RNA sequencing is becoming more and more common these days. I also briefly touched on those aspects in my presentation. I hope my presentation serves as a suitable talk for this conference, which focuses on the trends in biotechnology. So with that, I would like to thank for your patience. I hope it's not too long. I didn't take too long time, and I am open for any questions that if you may have. Thank you.
Thank you, sir, for your valuable speech. So we have been enlightened with the ribosome profiling and RNA sequencing. Thank you for that. The students might have got enlightened and be contacting you for further research areas. Thank you, sir. And the session is open for discussion. If you have any queries, you can ask the sir. Yes, students? Sir, it was a wonderful presentation. Again, I want to ask only one question from this presentation. Sir, is there any simulation method to study the function of genes so that we can bypass all this tedious knockout mice method? Because it's going to be a knockout mice method is it will take years to go. So is there any simulation method?
So first of all, knockout mice is not going to take many years. So when I started my PhD or before, when I was a master's student, for example, in Jamal, making one mouse knockout is a project for a whole PhD or a whole postdoc project. But the whole aim of the project, the initiative that I described in my first part of the talk, is to have the mouse knockouts ready for all the researchers that who are interested in a specific gene. So what they can do is they can get the mouse knockouts from these consortium it's a non-profit organization, meaning they're not commercializing their the mouse models. So you could study truly, if you could study the if you need to study a mouse knockout, you could easily get it. You don't need to make it. First, the second is, yes, there are a lot of simulation methods that you can access these days. So for example, again, the simulation method or modeling methods doesn't come just out of nowhere. So it uses the existing data sets. So if you wanted to analyze a function of a gene through a bioinformatics method, it always incorporates the data from the existing studies where they have used either mouse knockouts or any other model organism knockouts. So that's how the mutation studies becomes more and more important. So yes, there are a lot of to answer your question. Yes, there are a lot of simulation or modeling methods that you can easily use to identify. In order to do that, you should be able to familiarize with the available databases where you can get this information from. So one way to do is actually to look at the IMPC website that I mentioned, that International Mouse Phenotype Consortium, where they also have, so my skin example is one of the aspect of the project, but you also have data from other researchers like me who work on a different system, for example, the brain phenotype or musculoskeletal phenotype. So all these informations are gathered in one place for one particular gene, where you can access the data and then you can easily use it for your project, or for the questions that you may have in mind.
We are having another question.
Sure. I think the questioner is on unmute.
Sir, what is the role of pelota in embryogenesis?
That's a very good question. So our collaborators from Germany, they have shown that it affects the pre-implantation process, meaning that it does have a prominent effect on the cell cycle progression. So that doesn't allow the embryo to form. So that's what they have shown in that the first paper I cited. 𝗂𝗶𝗏𝗼
So
et
what is
al.
the role of
Reproduction.
Oh, okay, sir. What is the role of Wnt signaling pathway?
So Wnt historically been known as a pathway that is essential for development. So not only the embryonic development, but also many adult stem cell populations that are, for example, in the skin or in the intestine. So it is essential for, if you look at more specifically, generally it is for development, but more specifically in the stem cell development, stem cell proliferation. So that's where the Wnt signaling is important.
Thank you very much, sir.
Sir, I hope our students will contact you for all the queries. Thank you thank you so much for all the wonderful presentation. Thank you, sir.
Sure, no problem. Thanks for having me, and I hope you have a wonderful day ahead, and have fun and keep safe.
Now it's time for the tea break. Let's join back sharply by 11:30 a.m. for the second invited lecture. Thank you all. I repeat, now it's time for the tea break. Let's join back sharply by 11:30 a.m. for the second invited lecture. Thank you all. We welcome you back again. We are about to start the session. Some are born great and some achieve the greatness by their hard work. I kindly call upon Dr. 𝗕𝗶𝗁 Sir, Assistant Professor of our department, to introduce our invited speaker for this session.
Good morning, one and all. It gives me immense pleasure to introduce the chief guest of today's program, Dr. 𝗓𝗂𝗰𝗲 Chief Scientist in the area of Environmental Toxicology in CICR, Indian Institute of Toxicology Research, Lucknow. Sir has completed his MSc Botany in the year 1988 at University of Madras. In 1990, he has completed M.Tech Biotechnology in Anna University, Chennai. In 2009, Sir has completed his PhD in microbiology at Guru Nanga Deva University, Amritsar. He is now working as chief scientist and area coordinator in biotechnology system. This is CSR IITR Lucknow since 1990s. Sir has received a long-term fellowship award 1998 to 2000 for CSR DAA, and also he obtained short-term fellowship award 2007 for Indian National Science Academic INSA GFP General Research Foundation of Indian Technologies. Sir is a member of Genetic Engineering Appraisal Committee GEAC, Ministry of Environmental Forest and Climate Change. Sir has a greater technical knowledge in the field like microbial genomics, functional metagenomics, biocatalysis, and biodegradation. Sir has also done many project work on the in silico discovery on novel enzyme clinical expression suitable for microbial cost and identification of new biocatalysts by screening. SAR has undergone many training program, IPR, good laboratory practice, GLP, the safety assessment of the food derived from the GM crops. SAR also has the research experience abroad in the field of the molecular clinic and biosafety transgenic crops in the USA, Germany, and so on. SAR has published more than 21 papers, international journals. SAR was patent for the process bioremediation and contaminated soil. SAR has presented more than 25 conferences all over the world. We have such as eminent person with use of the right now. I am really proud to introduce him, I welcome you, sir. Thank you.
Very good, almost good morning. Dr. Gopalan, can you hear my voice? Am I audible to all of you?
Okay, sir. Yes, sir.
Can anyone confirm that my voice is coming?
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
Thank you. Okay. Thank you, Dr. Gopalan, for your kind words and nice introduction.
Thank you, sir.
And I must take this opportunity to thank all of you, the organizers, and particularly I see the lot of eminent scientists and dignitaries in this inaugural and program card, like Dr. 𝗕𝗏𝗿𝗁𝗲, Principal of the College and Coordinator of Department of Biotechnology, Dr. 𝗕𝗶𝗁𝗏𝗿, and of course large number of faculties and associated members with Jamal Muhammad prestigious college, and of course now I can say my friend who is coordinating the entire event with me, Dr. 𝗕𝗏𝗁𝗏𝗿. Thanks for this opportunity to share some of the research and the overall institutional activity what we perform, so that the students can benefit in coming days. So let me quickly come down to the talk. Later on, we can interact in detail for not only the research we do in a collaborative mode, also for some of the students visit, etc., etc. As I'm in pretty senior position, we can help out the students to take advantage of the research what we do, and of course the training and other skill set learning through Skill India Development Program. So all set? Okay, madam. Everybody is listening.
Yes, sir.
Yes, sir.
Okay. Okay. Let me share my slides. Yeah. Okay. So, can you see my slide in the screen? Yeah.
Yes, sir.
Hello.
Yes, sir.
Okay. Okay. Thank you so much. So, if Dr. 𝗕𝗏𝗁𝗏𝗿 is there, madam, how much time you have given me now?
Just 1 hour, sir.
1 hour. Okay. So it's 11:35. Let me try to complete before that. Well, my co-speakers also, spoke about very pertinent subject, like Dr. 𝗕𝗏𝗁𝗏𝗿𝗏 spoke about, I think, mice megagenetics, studying gene function at large scale, which is also a pertinent subject these days. How, like the genomic, proteomic, metabolomic things can help in understanding diseases or metabolic process. And I am speaking on microbiology, and one more speaker, Dr. Mukesh Thakur, will be talking about role of conservation genetics and wildlife conservation. As I see, there is a link between all these pollutant, metagenomics, microbiology, including even in their wildlife conservation, pertinent topics. So I think you made a conscious effort to bring people talking about current, really very pertinent to the theme of the conference, which is I think recent trends in biotechnology. So our work in IITR is on microbial diversity, metagenomics, of course, culminating all these capabilities to undertake bioremediation of persistent pollutants, which will reduce the environmental toxicant burden. This is our institute for the students. For the students' benefit, we are located in Uttar Pradesh, one among the few largest state, in Lucknow, which is the capital of UP, Uttar Pradesh. And this is our institute. There are several buildings like this with several center of excellence housed in these buildings. So just to give an idea, so our basic vision of the institute is serving it's like the motto goes like, safety to industry and service to industry and human health. So the vision for the lab is whatever the work we do should translate into societal benefit. in niche areas of toxicology. The major niche areas we focus is, of course, environmental toxicology, industrial settings. Of course, our institute started working with the help to industries and people who are working there, occupational settings, because for example, if we take miners, mining people, they get exposed to large number of airborne particles, where the pulmonary toxicology investigation is very important, like affecting the lungs, of course the inhalation route, etc., etc. So our institute started with the industrial and occupational settings, where we can provide them a possible, what are the routes of exposure and how to intervene to help for their health upliftment. Large number of publications and of course our work there linked to safety, where even after the miners come out every day, if they take a small amount of even the good, what you call jaggery, good here called in Hindi also vellam, so how they can clear off the lung depositions and particles. So we started with that, the institute, about 55 years back. But then we have a clear investigative toxicology, mechanistic toxicology, predictive and risk assessment. Large amount of computational tools, AI, like artificial intelligence, machine learning can help. And through those efforts long back, currently we have several branching out to address various issues. We, as I said, like system based, like all like is with neuro, starting from brain, liver, lungs, kidney. We have organ based toxicology, which is system toxicology, food and drug toxicology, which is very important domain, where our institute play a major role in analyzing food contaminants and of course the adulterants and toxicants there. Our institute is a referral lab, and it is recognized by FSSAI and regulated toxicologies. We are the number 2. Only 2 government laboratories are recognized for regulatory studies. One is NIPER, National Institute for Pharmaceutical Education and Research, which is at Mohali, Chandigarh, close to Chandigarh. But our lab also play a major role in undertaking regulatory toxicology, serving the industries, industrial product which need to be assessed for safety. So we have a good laboratory practice and National Accreditation Board of Laboratories. So we have those facilities, and we have large number of test organisms which are used under the alternative to animal model also to complement the animal studies. Yes, for addressing national needs like water, providing good water, good drinking water to people. So we have environmental toxicology, where we undertake mission mode programs like Ganga cleanup, Yamuna river mission, and various environmental aspects, like for example monitoring of air pollution, PM particulated matter 2.5, 10, 5, and the AQI, overall air quality index assessment. And also we undertake under this division environmental impact assessment of various industries, which includes thermal power corporation, telecom industries, automobiles. So we earn large amount. For example, every year annually about 20-25 crores we earn from this undertaking the environmental impact assessment alone. So we have translation research, of course, culminating all these technologies into, like for example, we have water device for providing a clean water, which can support the village folks from getting bacterial free water. We have developed devices. So we have a huge translational hub where even people can come and incubate their ideas and utilize our analytical and other facilities. We have that. Of course, we work for research for policy, recommending large number of guidelines, both in food and environmental aspect. We large number of our scientists serve in the national regulatory boards like CPCB and MOEF, DBT and DST. Of course, BAS, Bureau of Indian Standard Development. We play a large, large number of our scientists work there. Nanomaterial toxicology is our strength. We are the recognized center in national level. We do work on safety assessment and toxicology of several nanomaterial, be it like metal mediated or even like biological origin. We do have a huge facilities for nanomaterial toxicological implications to study the safety. Computational predictive, we have advanced computational facilities where we have big strength, and now the scientists and students working there, they put effort in addressing large predictions and pollution, structure protein relationship. So good amount of complementing the animal based studies with the computational predictive toxicology helps to large extent to derive information for acute, subacute, chronic toxicity, how these computational tools, machine learning, artificial intelligence can help. Of course, we do large number of surveys. We play a role in all these kind of national schemes. I will not explain, since our institute is located strategically here. We play major role. I myself was the coordinator for Namami Gange in the previous phase, and we still continue Namami Gange program, where Ganga cleanup is a major challenge and major work. We do quality assessment, etc. Of course, Make in India, Swachh Bharat, like Clean India, these are the nationally aligned scheme where IITR is playing major role. So all of you are there. Now I enter little more in technical talk, what we have done in the experiment in the laboratory based studies. Can I just say, are you all there? Okay, can I go ahead with the technical presentation now?
Yes,
hello.
sir.
Yeah, okay, thank you. So my own laboratory has worked on 3 major pesticides. These are very familiar to our Indian scientists, of course, and worldwide also. Gamexin, which is otherwise sometimes called as lindane, 𝗤𝗏-hexachlorocyclohexane. Generally, this is also called as hexachlorocyclohexane as 𝗕𝗶cie_H6. There is another famous pesticide. So all of you are located at south, close. Maybe many of your students are from Kerala. This Kasaragod district has this endosulfan problem, because endosulfan, another coordinated pesticide, sprayed on cashew plantation, that our mundri plantation, mundri seed. So this particular pesticide was found effective.
Excuse me, sir. Sorry for the interruption. Sir, we are having some disturbance in your connection, sir.
No, we are clear here, madam. I think someone to mute all others, please. You see, ours is a well standardized system. Nothing. I'm only sitting here and everything is okay. Just.
Okay, sir. Thank you, sir.
Everybody else, you see, so there is, I can hear a background noise.
Yeah. Okay, sir. Thank you, sir. You can proceed.
Someone else is getting such a problem, I suppose. Yeah. Just see, madam, in the lobby, you can mute others.
Yes, sir. We are doing that. Thank you, sir.
Okay.
Thank you.
Okay. Yeah. If such noise continues, just let me know. I can re-log in too. Yeah. Okay.
Yes, sir. It's now okay, sir. Now. Thank you, sir.
Okay. Thank you. Yeah. So, okay. These are the 3 major pesticides. Of course, this wash off from those Kasaragod, Western Hill residues reached the pristine waters in Kerala, and this is a worst environmental and aquatic, killing large number of aquatic organisms. Of course, DDT, I need not elaborate. This is heralded as wonder molecule, and the scientists who invented got large number of prizes, but the person who worked for its toxicity also recognized with close to Nobel Prize. So it's heralded as wonder molecule, but DDT once used stay for long, about 30 years in terrestrial soil, in aquatic system, little lesser years, but all these chemicals have been implicated in experimental model, and of course the samples have been collected from essence of different diseases. Like it is directly implicated to lead to neurological disorders, cancer risk. Of course, the endocrine disruption is well recorded. So lead to decreased fertility and of course abortions and reduced sperm count. So a lot of toxicological problems. What we do in our lab to eliminate such persistent chemical, we undertake biological, that is microbial degradation. I did not make more slide to explain what is biodegradation, etc., or bioremediation or biomineralization. application or employing bacteria to break down such toxicants are known as, simplifying the molecule is known as so this the breaking down of a complex molecule into simpler one and converting, if it is an organic compound, changing them to mineralization, like their carbon or nitrogen. So simplification of those molecules through microbiology or bacteria is biodegradation. Generally, if people achieve that through plants, it's phytoremediation. And these are the two major area our people work. But to look for a positive degrading bacteria, we go to a particular environment, do enrichment culture. If the information is known, we can directly derive those sequences to design a PCR or use it in southern hybridization. There are a large number of techniques. We worked extensively on microarray technology. We utilized all these techniques. Each one may need 1 hour lecture, so I am not going to load every information just today. Maybe another opportunity to talk to your students directly. I provide a case study to you. One of the chemical manufacturing facility which is very close to our institute is known as 𝗕𝗶𝗁 Limited. About 30 years they were making this chemical, and some of the residual chemical, I show it in next slide probably, maybe later on when I come. When they manufacture this chemical, which is like chlorinating benzene, it generates what I said, 𝗥𝗲𝗰𝗲. When they produce this chemical, it generates 4 isomers. Only one of the isomer, about 15 to 20%, has the insecticidal property to kill the insect when they spray into an agriculture field. Rest of the chemical is known as a muck. As you can see, large amounts of mucks were, like pushed out of the industry, and this canal running, you can see the flakes of chemicals. This is illegal dumping of this chemical. But as I told you, these are, highly toxic chemical. This canal falls into a river called Gomti in Lucknow, which ultimately feeds river Ganges, making the problem more serious. This pesticide, as I said, chlorinating benzene is so simple. It was cost-effective chemical throughout the world they produce. As you can see, like even developed world, like former European countries were not so developed like India, and they do all this chemical in open dump. I have visited one of such sites in Germany near Gnitz, where such chemicals are now put into salt mines and they are sealed it, so they are not in open dump now. So we go to an environment such is like for basic students learning. I put this slide. You go to a contaminated environment, collect water samples, sediments, or a rhizospheric soil, and bring into lab, and then you enrich, what you call enrichment process. You can enrich that and inoculating the total soil as a starting inoculum. When you do see, when we immediately plated, you can see large number of diverse bacteria appearing. But for example, if you enrich with a particular substrate, this we have done it with polychlorinated biphenyl, such a diverse bacterial load lead to adapting into this chemical, which can break down, utilize them as a carbon source and utilizing them as a sole source of energy and carbon. They can only multiply and survive. Can also detect some of those bacteria, able to dechlorinate them. As I spoke about chlorinating compound, simple dye can indicate. I don't want to go into too technical, otherwise you will get bored. So what I'm trying to say, there are dechlorinating mechanism which will convert phenol red into orange color. That shows that there are free chloride removal by an enzyme called dehalogenase. You can catch hold of that bacteria, and also when this bacteria is spreaded and sprayed with some chemicals, you can see the like when we do microbiologists antibiotic susceptibility test, it gives the clearance around the colony. So you can see this nice even single colony bacteria is mineralizing those compounds. So we pick up those bacteria, and large number of pathways we study in our lab. We have extensively worked on enzymes, which are when a specific enzyme act on this compound is known as upper pathway enzymes, and after protect-attenuate, catechol, and other pyruvate, say protect-attenuate, catechol, 𝗕𝗔. These compounds act as a central intermediate, and then when the compound is cleaved to serve as a energy yielding intermediate, they are called lower pathway enzymes. As I said, those enzymes can be exploited. For example, monooxygenases and dioxygenases play a major role in aromatic compound degradation. What we do, we go to different environment. For example, this bacteria we have collected from Ganges river water, and after plating, we provide indole as a substrate. Then it turns, because of aromatic oxygenase, it turns into blue color. You can pick up those bacteria and export them in pure culture. As I told you, we go to different environment. I had a project to look at the bacterial diversity in contaminated ecosystem. This is like God's own country in Kerala. Hindustan Insecticide Limited, founded. This was the one among the first chemical industries founded by Nehru, because we wanted to agriculture or food sufficiency for our own folks, our own countrymen. So fertilizers and pesticide industries were one among the initial industries developed by those governments, and the industry generated large number of effluents, and then this falls on to many of nowadays there are more stricter policies by NGT like National Green Tribunal and other pollution control board. But earlier days, these effluents falling directly onto the river, and then river falls onto the Arabian Sea. It's a huge issue there. We collected some of the sample, isolated. You can see, 𝗕𝗶cie hexachlorocyclohexane, large amounts of metabolites of DDT. These were detected in those soil when we have collected there. Other than that, countrywide we have large number of such superfund sites. Like many of you from Tamil Nadu know that even Noyal River near Erode, this is a huge Karur that falling onto Kaveri River, and people because of this textile dyeing and coloring of cloths and threads, people have dug well about even maybe 20-30 kilometers. They have seen the colored water coming out. One such major problem near Noyal River area. But like that, countrywide 136 priority polluted sites are there, which is identified. I am part of this National Coordination Committee under Central Pollution Control Board. So these are the priority sites which have been allocated for remedial aspect, be it engineering approach, biological approach. This has been designated to be acted upon quickly for sealing, not contaminating those lands where people have affected severely. So the chemical I explained, I am coming back to one particular word. This chemical hexachlorocyclohexane produced in large amounts, and these bacteria have been identified, worked on these isomers, as I said in this slide, like alpha, beta, gamma, delta, all of them are toxicants, and then the bacteria is able to grow clearly and degrade them. And we identify any particular biodegradation has to be authenticated and supported with degradation, of course, simplification of the compound and complete mineralization, identify the metabolite, of course, the protein, so that proteins and enzymes and genes involved, so that we can elucidate the complete degradation pathway, which you can see in the next slide. Like there are several groups in the in world, one Japanese group, French group, and our own in laboratory, and in Delhi one more, Professor Ruplal worked on these chemicals. We have elucidated this pathway completely, identified the genes. Moving from degradation, I will come back often and on. There are many things can help in when we go to the field. For example, all of you know, many of you are all working in biotechnological, microbiological, or biological laboratory. Associated things like bioattenuation. biostimulation, biomineralization, of course, biosurfactant. When we go to the field, these chemicals are strictly or tightly adsorbed to the particle, soil particle. Sometimes they percolate down to the earth, but because of their adsorbent, they are not available to the bacteria for degradation. And they are hydrophobic too. Because of these properties, their persistence is usually for longer period. So we isolate biosurfactant, which can desorb and help in bringing these chemicals, and we produce them in large scale. For example, we bacteria, we identify whether they are having biosurfactant or not with several methods. This you may be knowing literature. Then we extract the biosurfactant and identify what is it through all FIR and other analytical techniques, and then take them back to the field where this chemical. I'll show a case study where we have employed these kind of chemicals for desorping these desorption of this chemical to make them available to bacterial degradation. Yeah, we work on flask and then of course identify, extract them, purify them, and we in our own lab we have a 7-liter fermenter. Up to 4 to 5 liter we can aerobically grow these bacteria, and we produce them in large scale, or take these cultures, sometimes add to the soil where we undertake the remediation work. Some of these bacterial surfactant, as you are all biotechnologists, can be exploited for additional applications, and other than the remediation work. We found that some of this biosurfactant can act upon inhibit the growth of 𝗕𝗶𝗁𝗂, Klebsiella pneumoniae, Pseudomonas aeruginosa, of course, E. coli, Bacillus subtilis, and of course this Aeromonas hydrophila. Some of these pathogens can be inhibited at different concentration and killing them effectively, where this can be further taken into formulations of antiseptic. There are a large number of companies have worked on it, but we are also moving into patent some of this surfactant and their process associated with it. Coming back to the application, which most of the students or maybe faculty is interested, one of the site I explained about industry, India Pesticide, which is located in Lucknow, has illegally dumped those chemicals in about 3 to 5 acres of land and completely contaminating the 7-8 villages surrounding there, because these mucks were dying and people are utilizing them in poor farmers spraying them into potato crop. So we have taken a project from Department of Biotechnology to remediate that site in an organized manner, involving engineers, plant biotechnologists, microbiologists, industrial, chemical engineering people, all joined together. There is about 4 crores of amount funded by DBT. We have prepared the site. As you can see, about 3 to 4 meters of this chemical has been pushed. First, they taken to the top layers and then dug out and prepared this, under the US EPA guideline, Environmental Protection Agency guidelines. And then we brought to the field like this. You can see a nice field prepared, bringing back the chemical, and then we brought these kind of bacteria which are able to metabolize them, and in a cold chain from a large scale fermenter, about 50,000 liters every day, producing these bacteria added into carrier materials, and then sprayed back into the field, so that such bacteria also exist there, but we grew them in large scale, augmented them so that the degradation can be facilitated to go in a faster track. As you can see, the field which is like in the upper panel like this, and we have employed this is like 𝗕𝗶 facility, the Energy Research Institute at Delhi. We grew these bacteria and packed and brought it here. Okay, as you can see the action here, we planted large number of plants which have been growing in that region. Phytoremediation on field. We have divided the field into many experimental sites, like only bacteria, only plants, and plants and bacteria, and some of them are control sites. You can see the action. Very, very interesting. Laboratory based work, how it can take see the end of the tunnel of the research going into the field approach. This is Professor 𝗔𝗶𝗄 himself spraying the bacteria onto the field. So This is me who has initiated this, this field trial here at Lucknow. Similarly, I have worked on, I will not go into detail, I will move fast now because similar, same approach. Isolate bacteria, identify metabolite, grow them into large scale, identify the compound which can degrade. I have 6 more bacteria which made into consortium for oily sludge. I think as you are all from Tamil Nadu, I know that you will be aware of there was a spill. There is a ship called, I think, some Kanchivaram, and one more ship collided near Chennai Ennur coast. I was traveling, as maybe many of you might have read my profile. I am originally from Salem district, so I was traveling. At the time, I heard that there is a spillage of oil there. So I just took a taxi, went there to the coast, and interacted with the people who are working there. And as you can see, large number of tortoise, fish, death, and of course, this is one particular small ecosystem affected. But all of you might know that Prince Alaska Exxon Valdez spilled several times. Every now and then there are ships releasing crude oil into marine ecosystem. And marine ecosystems are one which are threatened due to this oil, especially crude oil, which has large amounts of carcinogens, asphaltanes, alkanes, alkenes, polyaromatic hydrocarbon like benzene 4 and above ring compounds cause cancer directly. And silently many of these fish and marine species, planktons, flora and fauna getting endangered. This is a very serious threat. I saw the people who have taken the contract, some company, they were like using hot water to wash off this. You can see this slide, how this is like, affected, and then just washing off, like letting them, the oil go back to the sea. Large number of other possibilities are there, but they have followed this. Anyway, so with that technology, we have also undertaken a work at ONGC Ahmedabad. We are getting some of the crude sludge generated, by ONGC. It's a huge amount of such sludges are generated, and there are storage field where we are undertaking engineered and of course biological approach with it and treating them for the safer, reducing the pollutant there. Our institute has, of course, these alternative to animal model for ecotoxicity risk assessment organism. Other than honeybee and this bird, which we may be establishing soon, we have earthworm for terrestrial toxicity, algae, daphnia, and zebrafish. These are all there, and one of our scientists, Dr. 𝗓𝗏 is helping in our project. He is also from Tamil Nadu near Trichy only, Perambalur. He has established this facility. He also works in this regulatory toxicity, utilizing this organism for safety evaluation of large number of industrial products. Because when we do such assay, ecotoxicological risk assessment is very important. So we do endpoint assessments of chronic toxicity exposure, acute toxicity, LC50, LD50 determinations, and we do perform long-term assays, of course, the reproductive toxicology here, like cocoon formation. Anybody working in the earth, behavior, starting from the behavior, which I'll show it in next slide. For example, if you throw earthworm into contaminated soil, within few minutes they try to move into a non-contaminated soil and then get burrowing habitat, safe. And then we, starting from behavior, we go up to cytotoxic, genotoxicity assay, reproductive assay, and then we do this assay during the different stages of biodegradative experiments when we undertake the field work, because different time how much compound is reduced, we do it in the analytical, of course, using gas chromatography, etc. But also we side by side do this earthworm or maybe other organism based earthworm, zebrafish for aquatic toxicity. How is these chemicals are still affecting to finally lead to site restoration. So I'm moving on to other area. So all of you are there. Is it interesting, or all of you are want some break? Hello. Hello, Dr. Gopalan. Madam,
Yes, sir. It's
Mafia,
very interesting,
Mohideen.
sir.
Huh?
Can continue, sir.
Yeah. Can I continue? Yeah. Hello.
Yes, sir. We can go ahead.
Okay, madam.
Yes, sir,
Okay,
we
thank
can
you.
go ahead, sir.
Sure. No, I just wanted people to be alert or maybe some break. So what I want to explain, all of you must be reading in newspapers, and it's in environmental news, and large number of projects are also being sanctioned by Government of India. We have in CSIR, Council of Scientific and Industrial Research, we have something called project theme-based projects. Theme-based projects which is known as ecology, environment, and air. Recently, what I did, large number of work happening in small pockets throughout India, and of course also Western world. People are working for something called trophic transfer. Plastic is as such a problem, and also the microplastic, the fragments broken into pieces less than 5 mm of these reach various compartments of the environment, marine, terrestrial, and large. All of you know that large number of dumps. Global waste generation of plastic is huge. All of us, even for buying a 5 rupee things to a a industrial container, various like polyethylene terephthalate, polyvinyl chloride, polystyrenes. There are 6 major class of plastics. Of course, each one of them break down to various their intermediate monomers, polymers, and create havoc. And many of these organisms in the trophic food web accumulate them and pass on to next food web, in the higher order. Of course, in marine, it's very huge. But let me take you some case studies where we have been associated a little bit. So what I told in the team CSIR, we proposed a project with 7 institutes. I am the coordinator for that. We will soon embark into these kind of huge dumps, and then work on the leachates. What are the chemical leachates coming out? What is the impact in the surrounding ecosystem? As I am coordinator in the environmental toxicology, we have huge responsibility, as our institute also works on toxicology coupled with it. So you can see, this is near Ghazipur in Delhi. If anybody travel from Delhi to Chandigarh, Karnal, and Punjab side, they will pass very close to this mountain of plastic, municipal dump. Having, of course, you can see the close picture of this is a water body which is running close. All people even take bath, and there are a lot of activities happening there. If anybody is from Chennai, will know that Perungudi and Kodungayur. I visited this site, because I have a small project with the NIOT, National Institute of Ocean Technology, near Pallikarnai. So I go there and I saw this. Now people have government initiative from Tamil Nadu. They moved these sites, and they have taken it out, and some of them are treated containments. They have made it better now. I must appreciate that effort. But this lake, these are all lake adjacent to coastal regions which have been dumped like this, and it creates large number of ecological disaster and implications. And every city, major cities like Mumbai, and if there are students, in our age, I tell you, in movies also and in science magazines and even textbooks, we read about coral reefs. This will get extinct. Many of them, these huge coral reefs are going to die by 2050. This has been predicted, scientifically proven. So it's a huge problem. Madam, are there students there in this PhD students and maybe postgraduate students are there part of this conference other than faculties? Hello? Hello?
Yes, sir.
Ma'am, I just for a couple of minutes I will go into a video mode for just 2 minutes. राजमा प्ले होगा, ये पहले भी नहीं हुआ, लोड हो रहा। हाँ पहले चेक करना चाहिए. Give me a minute, please. Just a minute. Can you play? Okay. Can you play this? Open. What? Ma'am, can anybody confirm? Are you seeing a video running?
Yes, sir.
Yes, sir.
Just for a couple of minutes I will play. You can also just relax, and maybe some students they will find it interesting, but people can just Google out and YouTube. I'm just giving an implication how we have threatened this coastal ecosystem, and how this future generation will see marine organisms and coral reefs. This is near Bali, Indonesia, and you all know maybe this Bali is attracting large number of tourists, and this is the underwater when they come out. Can you just for a minute they can relax and watch this video. I have 15 minutes more. Hello.
Yes, sir. Yes, sir.
Ma'am, do I have maybe 15-20 minutes, or how much time I have more?
So you can take your time, sir. No issues.
No. Maybe just in 15 minutes I will conclude.
Yes, sir. Thank you, sir.
Yeah. I'll stop sometime in between this. Just to give how this marine ecosystem is affected because of plastics, and of course when they break down into microplastic, these are ingested by large number of marine living organisms, and then now this plastic is very well characterized, which takes up other chemicals as an adsorbed mechanism, and this again magnify the problem into manifold. So this is really a serious issue which needs to be tackled. That's why global efforts are underway for plastic and microplastic elimination, banning of plastics. We need to play a major role there. Yeah, I can stop. So sometimes the students can. Okay, I'll stop and come back to the talk. Yeah. Close. Okay. Okay. Just a glimpse of that. Was a disturbance, and then bisphenol is a problem. Pollution effect on marine animals well known. Some of this effort, these plastics in our own sister laboratories at CSIR, IIP, Indian Institute of Petroleum. We work on toxicity assessment of some of these aspects, but then this plastics has been converted into gasoline, diesel, and of course some of the aromatic chemicals. This is the technology developed by our own CSIR laboratory. So one of such site is very close to Lucknow, where our institute is located. This is a municipal site, like I showed it in other cases. This is the leachate coming out, and then I just took a sample out of this. There is a river running close by, which I said 𝗕𝗶𝗿𝗁 river, which is a perennial river. As you can see, this nala, what you call drain canal, carrying this toxic waste falling on directly into river. It is again huge issue. So we are screening, taking some samples, looking at the bacterial population, and taking them to degradation of various plastic intermediates. We are currently working on their adherence and to the polythene and polythene surface. We look at it, and of course characterize those bacteria and like enzymes, hydroxylases, PETase, and lacases. Large number of enzymes present in such bacteria for breaking this kind of plastics. Of course, there is one beautiful robust bacteria has been reported in Japan. That bacteria is identified as 𝗔𝗔𝗌 𝗔𝗂𝗃ensis. The scientist is some Sakai, and professor, he clearly delineated mechanism of PETase, how they break down PET bottle, polyethylene terephthalate, like our own, like Bisleri or Kinley bottle, adhering onto them, forming a biofilm and making them fragile and converting those compounds into like simple molecules like ethylene glycol and 𝗕𝗏, which can be again monomers can be exploited for synthesizing or maybe other energy compound. This is the bacteria they have identified. We did also ventured into metagenomics, where we have taken sample from 4 different rivers. Metagenome, I don't want to go into detail. Each one needs one lecture. So metagenomic, just the entire genetic material of given environment. It may be a ecosystem, maybe a gut microflora, or your oral cavity, or skin metagenome. There are, addressing the entire or studying the entire genetic material and DNA directly without cloning. And we can also make bigger libraries, cosmid and phosmid libraries, but we can study them through various sequencing platforms. We did that and collected large number of samples from various Ganges river locations, looked at their culturable biodiversity. Of course, metagenome extracting sample, like this sample we collected from Upper Ganges near 𝗕𝗶𝗿𝗁, subjecting to sequences, and then exploit them for antibiotic resistant genes, bacteriophages, which river, how much abundance, which sample, what are the antibiotic resistant genes for which class of antibiotics. So these kind of detailed analysis. This slide can be split into one big lecture. So because we have identified phages present there, acting upon some of these antimicrobial resistant bacteria, which we call it MDRs, multi-drug resistant organisms. Some of the bacteria has more than resistance, more than 20 antibiotics. They are like super bugs for MDRs. So we have detailed studies for each sample locations. So the key is like, you have to educate the younger generation, younger populations about prevention is better than cure, of course, not to use, but then what are the implications of such toxicity by dumping these chemicals? If nothing is available, especially for the plastics, let us actually Tamil Nadu can teach others. Come back to what we call, if the students are there, they may just laugh out. Come back to this what we call manjappai. We நாம முன்ன இருந்த பீரியட்ல இதுதான் கடைக்கு போறது எல்லாமே, everything we did through this only. இப்ப யாரும் இந்த இது ஃபாலோ பண்றதுக்கு கொஞ்சம் கூட பிடிக்காது மக்களுக்கு. எல்லாம் பேக் வித் ஆல் பிளாஸ்டிக் மாட்டிட்டு போடும். பட் ஐ திங்க் ஓல்ட் இஸ் கோல்டு. நம்ம ஓல்டு டைம் தான் பெட்டர்னு நான் இன்னும் நினைக்கிறேன். ஸோ ஐ திங்க் வி ஹேவ் டு கம் பேக் டு திஸ் pattern now. Okay, I'll stop here because I can speak on each aspect for another 1 hour, but let me conclude here by saying large amount of pollutants are generated every time. We need to find out like benign by design, green chemistry approach, alternatives to these chemicals, and safer chemicals for even agriculture or biocide, moving to organic. But will that be sufficient? Can we still going to alternative to pesticide? Can we meet the end of the food production? All these things are debated. That's why we are impacting, slowly moving into organic, alternative to these chemicals, or less persistent chemicals. All these are being addressed by various groups of scientists, including the regulatory bodies and the regulators. Different.
Sir? We could not hear you, sir.
Sir, please unmute your mic, sir.
Excuse me, sir.
Sorry. So yeah, just what I concluded is that our group has put effort in addressing various issues in environment, working on biological solutions for remediating some of the chemicals, risk assessment, and some of your students, PG students or research students, anytime want to have some interaction, they can visit. Of course, maybe once the corona time improves slightly better. Other than that, I will be happy to visit sometimes your university, your college also. So with that, I will conclude. But if some of you want some curious questions, I am open for next 5 minutes, so that then we can stop. And I do have certain work to attend to. Thank you, madam. Thank you so much. Thank you, all of you. And thank you, dignitaries, eminent scientists who are all present there, and those who joined through the virtual mode. I thank for your effort in listening to whatever I shared. So maybe the organizers can take over from me now. Thank you.
Thank you, sir. If you have any doubts, you can ask.
Students, you can interact. The session is open.
Yeah, some curious question.
Wonderful presentation. Thank you for that. Sir, I have one question.
Please.
Sir, is
May you know
IITR
who is asking that question? Can you introduce?
Sir, this is Dr. 𝗕𝗶𝗁𝗲 from 𝗕𝗶𝗿𝗁 College.
Yeah, you are there, yes. Yeah, please go ahead, madam, please.
Sir, the question is that, sir, I just want to know, is there any bilateral consortium you can provide commercially to other NGOs and workers so that they can replenish the environment near to them?
Okay. Yeah. Okay. Thank you, madam. Thank you for that question, because that's also pertinent to application. The two technologies, the consortium we have developed is now with the NRDC, what we call National Development Corporation Research, NRDC, National Research Development Corporation, which operates under Delhi IIT. So we have outsourced, or we have given this know-how to them. Of course, if anybody want to undertake this, can enter into some kind of MOU and some kind of license fee we may ask, and through our planning people, then we can think about it. But of course, as a scientific outcome, this should be replicated into various area where we can do this application. But we need to get into some kind of understanding. Then it's available. Yeah, sure. For to answer you, yes, it is available. But after certain official processes where we need to enter into an agreement, both for NGOs and maybe an organized government institutes, or maybe even industries, we are open to that. Yeah, sure.
Sir, can you share us any public participation, so that is there any public participation in replenishing their land or like that? Any?
Yeah. Actually, as you said, a large number of people, small MSMEs, medium industries. For example, there is a group called Ramke in Hyderabad. They are but industrialists. What you are asking is public group. I think not many public group joined hand. The like NGOs or maybe a local community who is undertaking. They are all working on the preventive mode. For example, the field we are treating, it's only a scientific, proof of concept approach we are doing. But public participation, NGOs, and there are large number of groups which are, they are there. In Mumbai, for example, whenever I go to, in Delhi IIT, we have a soil network group. There are large number of companies come there who are doing little bit in this, but I don't know their details. They from NGOs or public group, but they do play a major role as an environmentalist. I know that the organized group is undertaking such major work. Yeah.
Sir, thank you so much, sir. Thank you for all answering.
Nice question. These are application oriented. Yes, thank you, madam. Any more questions, please? Anybody? Any students? Anyone of them? Are there research students who want to ask some doubt?
Sir, good afternoon, sir.
Yeah, please. Introduce yourself.
This is Dr. 𝗕𝗫𝘁𝗵𝗵, sir, Assistant Professor of Biotechnology, sir.
Yeah, yes, please go ahead, madam.
Sir, in microbes, they specifically cleave the 𝗕𝗶𝗁𝗲𝗄𝗏₼, sir.
Yeah.
Is there any specific mechanisms for dechlorination process, sir?
Okay, clear. Good question. Technical question. I appreciate. See, as I told you, there is a dehalogenase or dechlorinases. Because as I told you, this compound, no, HCH, hexachlorocyclohexane, C6H6Cl6. The chloride, you know, HCl, you know, the bond is cleaved off by the dehalogenase or dechlorinase enzyme. If you punch even my own paper, lin genes, LINA, lin A, lin B, lin C, you know, such lin D, there are genes we have reported. These lin genes are very unique enzymes, act upon those molecules, successive dechlorination process, they knock off this chloride molecule, forming HCl, and then removes this return, and then the dehydrogenase takes over. Then see when they form hydroxyquinone and chlorohydroxyquinone before that, then the lower pathway enzyme I explained you, like mucinate isomerase and then Krebs cycle intermediate. So upper pathway is dechlorinase enzymes, then dehydrogenase enzymes, and then the ring cleavage enzymes, which are dioxygenases, monoxygenases. So this is the successive dechlorination mechanism. Good question. Thank you. You look at the papers. My own articles are, there is another author called Nagata, Nagata, and then lin genes. This is a very, very good mechanism established and very beautiful mechanism, because many of these enzymes are very unique, not reported earlier. So it's a beautiful enzyme system where some of them we cloned and purified protein, and protein structure also we looked at it. Thank you.
Okay, sir. Thank you, sir.
Okay, nice question. Yeah. Anybody else? Just two more questions we can take and then close. Yeah. Hello. Organizer. Anybody? One more question, maybe?
Hello, sir.
One doubt?
Hello, sir. This is Dr. 𝗕𝗫𝘁𝗵𝗵.
Yes, ma'am. Please go ahead, madam.
What is the mechanism of 𝗕𝗫𝗁 enzyme? Where it is present, how it is isolated, sir?
Yeah. Actually,
what is the chemical name of 𝗕𝗫𝗁? Chemical name of 𝗕𝗫𝗁.
See, this enzyme is polyethylene terephthalate only. Its protein structure, everything is elucidated. This is a sort of membrane-bound enzyme protein from this particular, I said Idionella sakaiensis. There are other proteases which is poorly studied. Okay, so as I said, membrane-bound, and it acts upon this polyethylene terephthalate, converting them to monomers. As you could have seen, that they convert them to terephthalate and ethylene glycol. These are the monomeric compounds, which of course can be, as a raw material for other things. So this is an interesting enzyme. That the chemical name itself is polyethylene terephthalate enzyme. Okay. Yeah.
Okay. Thank you, sir.
Yeah, sure. Any more question, madam? Anybody? Otherwise, I am done. Maybe you can close from organizer side. Maybe you can move on. Yeah. I think I'm done.
Yes, sir. Thank you, sir, for giving us the valuable insights.
Your voice is breaking, madam. Not audible. Hello? Yeah.
Thank you, sir, for giving us the valuable insights.
Okay. Thank you so much for this opportunity. Can I close now? Yeah. Thank you, each and everyone. Yeah. Thanks. Bye-bye.
Now the time is approaching for lunch, and let's all assemble sharply back by 2 p.m. for the second technical session. We have an announcement for the participants of paper presentation. The presentation should be in the PPT form. Allotted time for the presentation is 5 minutes, and 2 minutes is given for discussion. The participant should be present here sharply by 1:45 p.m. Thank you all. We welcome you back again. Shortly, we will be starting the second technical session, that is the paper presentation. We hope the participants of the paper presentation are ready with their presentation. I kindly request all the participants to enter your full name in the Zoom meeting.
I repeat, welcome you all back. I kindly request all participants to enter their own full name while entering the Zoom meeting.
Here's an announcement for the presenters, participants of the paper presentation. I kindly request all the participants of the paper presentation to enter their full name in the meeting.
Welcome back. Thank you all again for sticking to the time. Let's move on to the paper presentation session. We have Dr. 𝗕𝗶𝗁𝗲 ma'am and Dr. 𝗓𝗏𝗫𝗓 ma'am, assistant professors of our department, as the evaluation judges for this session. The winners will be announced at the valedictory event. Now, I request the judges to take over the session. Please, ma'am.
Good afternoon, participants. Your allotted time is 7 minutes, 5 minutes for your presentation and 2 minutes for discussion. So conclude your results within the scheduled time. Now I call upon C. Mohana Sundari, Muttayamal College of Arts and Science, to present the paper.
Yes, ma'am. Ma'am, shall I start?
Yes, ma'am.
All of you view my PowerPoint?
Yes, ma'am.
Yes.
Okay. Good afternoon to all. First of all, I express my thanks to our institute giving me this best opportunity. Thank you, ma'am. Thank you, sir. I would like to present my paper on antimicrobial activity of 2 medicinal plant extract against multi-drug resistant Pseudomonas aeruginosa. Antibiotic resistance is the major problem which increase morbidity and mortality. Resistance also leads to treatment failure. Based on this above fact, I have screened 125 pus samples from secondary care hospital in various sources like IPD and OPD. Among the 125 pus samples, 65 samples found to be positive in various pathogenic bacteria. Out of these 65 positive samples, 4 multi-drug resistant Pseudomonas aeruginosa were isolated and identified and isolated and screening like. These 4 isolated resistant to maximum number of antibiotics like amikacin, chloramphenicol, tetracycline, cifotoxime, piperacillin tazobactam, astronome, ceftazidime, cefepime, imipenem, meropenem, tobramycin, ceftriaxone, zendamycin, cifoberazone, ciprofloxacin, amoxicillin, ampicillin, cotrimoxazole, and levofloxacin. Since most of the antibiotic found to be resistant, so that I have selected 2 medicinal plants for this study. Like one is the 𝗕𝗏𝗁𝗲𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏
𝗕𝗏𝗏 and the genus மெடராஃபெட்டன்சிஸ். Tamil name is 𝗕𝗶𝗁𝗁 or holy mookan. My second plant is 𝗕𝗶𝗁𝗁 𝗂𝗍𝗓. The genus 𝗂𝗍𝗓, the family 𝗕𝗏𝗏𝗍𝗔𝗍, the genus 𝗕𝗔𝗏𝗁 and the species 𝗂𝗍𝗓. Tamil name is சூரைமுள்ளு or சூரை இலந்தை. This mainly combined study of these both medicinal plants having the wound healing property traditionally. Combined effect of two medicinal plant extract. I have used the solvent like benzene, methanol and ethanol were analyzed by agar-well diffusion method. Two different concentrations were used, 50 microliter and 100 microliter. 50 microliter concentration of extract showed 15 to 17 mm zone of inhibition, whereas 100 microliter of concentration of extract showed 18 to 24 mm. The results is showed in this tabular column. The conclusion of my study is based on the result, further phytochemical studies to be carried out to find out the active components of 2 medicinal plants inhibiting multi-drug resistant Pseudomonas aeruginosa, which will be useful in the information of drugs. Thank you. Thank you, ma'am.
Hello, Mohana Sundari.
Ma'am.
Nice presentation, ma'am.
Thank you, ma'am.
You have used methanol, ethanol, and benzene solvent extraction.
Yes, ma'am.
Why can't you use aqueous extraction for your antimicrobial activity?
So, ma'am, sometimes aquatic is having some contamination, ma'am. Some fungal contamination is a major problem in my study. That's why I was already I examined, ma'am. But finally, I have selected the benzene, methanol, and ethanol, ma'am.
Which solvent extraction is best one among benzene, ethanol, and methanol?
Ma'am, mainly methanol is the best one, ma'am, in my study.
Why? Why it is so?
It's mainly its extraction is very pure, and same time, ma'am, the solvent, the powder form is easily derived, ma'am.
What are the phytoconstituents present in your plant? First one and second one plant.
Ma'am,
phytoconstituents.
Madam, ma'am.
Phytoconstituents, secondary metabolites.
Ma'am, it's my studies is going on, ma'am. And this is a support of study.
Okay, what are the positive and negative control you use for antimicrobial activity?
Ma'am, positive control, I have used the direct plant extract, ma'am. Directly the solvent, ma'am. Solvent, I have used the solvent.
It is a negative control. What
yes,
about positive control?
Positive control, I have used the plant extract, ma'am. Pure plant extract, powder
is
form.
Sample. Your sample,
yes,
no? Plant extract is your sample.
Yes,
so positive control, you use any antibiotic.
Yes, ma'am. I have used the mixed antibiotic, ma'am. Broad spectrum antibiotic I have used, ma'am.
Okay, ma'am. Okay. Yes, thank you.
Thank you, ma'am.
Ma'am? So which organisms are most sensitive to this medicinal plant? Which organism? Pathogen? Okay, thank you, ma'am. Next, lot number 3, Reem Jalaluddin, Jamal Mohammed College, Trichy. Lot number 3. I again repeat, lot number 3, Reem Jalaluddin, Jamal Mohammed College.
Hello, ma'am.
Yes.
Am I audible?
Yes, ma'am.
Ma'am, I'm not able to connect my presentation, ma'am.
Sure.
Ma'am? Let us look on. Ma'am, I'm there in two names. So the other Reem Jalaluddin is the one from which I'll be presenting.
We have shared the PowerPoint dream. Is it visible?
No, it's not visible. Your screen is visible, but my presentation is not visible.
Ma'am, we have given the permission to you. You can share your screen.
Okay, sir. Thank you. Ma'am, is my screen visible?
Yeah, visible. You can continue.
Thank you, sir. Good afternoon. My topic for today is about anterior cruciate ligament tear. If we see, anterior cruciate ligament is a ligament which is present in our knee joint. And knee joint, it is the joint which is affected the second most after the ankle joint. So what is anterior cruciate ligament? So can you see my slide?
Yes,
Okay.
ma'am.
The bone above is femur, and below we have the tibia and fibula, the sisterly bones. The anterior cruciate ligament is a ligament which arises from just behind the lateral condyle and ends in the medial meniscus. Over here, can you see my arrow? From lateral condyle to the medial meniscus of the tibia. So let me just give you an overview of the ligaments present in our knee. There are four ligaments. One is the lateral collateral ligament, medial collateral ligament. These ligaments prevent our knee from moving in the left and right position. Anterior cruciate ligament and the posterior cruciate ligament, these two ligaments have similar properties and prevent the femur from moving forward and backward. I mean sliding off. They help in moving in 90-degree position, but prevent from sliding off the tibial bone. The anterior cruciate ligament and the posterior cruciate ligament, both of them have similar, they have similar composition. Medial collateral ligament and the lateral collateral ligament have similar composition. The main difference between the anterior cruciate ligament and the medial collateral ligament is that the anterior cruciate ligament has synovial fluid surrounded by its ligament, whereas the medial collateral ligament and the lateral collateral ligament do not have synovial fluid. Medial collateral ligament has blood supply in its ligament, but then the anterior cruciate ligament and the posterior cruciate ligament does not have blood supply. This is indeed a very big drawback because MCL and ACL, we just tell it in short form as ACL and MCL. ACL, these are, or when they begin to tear, automatically in MCL, the blood oozes out and begins to clot. Once it begins to clot, this gives time for the collagen production and regeneration. Therefore, it leads to healing. But when we see an anterior cruciate ligament, as soon as it begins to tear, what happens? The synovial fluid moves in the middle and does not give us the opportunity to heal. And moreover, this does not have blood. Therefore, there is no clotting and no healing. So we are to use clinical methods for regeneration and reconstruction. Up to now, the procedure which is used is that we take a graft. The graft is mostly taken from behind our thigh, from hamstring muscles, the posterior part of our thigh. The hamstring muscles are taken, and a hole is drilled from the tibial meniscal end to the lateral collateral condyle of the femur. And then the graft is inserted and screws are screwed on both ends. This takes around like 6 to 8 months to heal. This is actually not successful in 100%. This is not 100% successful. Golden standards say that it is successful up to 80%, but as per statistics, it's almost only 40% success rate. This is just a comparison between the classical and tissue engineered approach. Classical is that we just take a graft, that is autograft, from our own thigh and insert. And tissue engineered is that we reconstruct a scaffold outside and then insert it inside. So these scaffolds are, many are used, but I have selected a few which are really successful. First is 𝗓𝗏𝗏𝗏. 𝗓𝗏𝗏𝗏𝗏, this is actually, it is quite a long and lengthy process, because in the initial stage, the 𝗓𝗏𝗏𝗏𝗏, it has toxic protein called serine. This serine has to be removed, or else it will inflame our body. Therefore, it is removed. And once the silk is removed, fibronin, which is this compound, is really very tensile. That is, it will allow us to be, it will allow our knee to be flexible, and it is biocompatible. That is, it will, it does not lead to, what to say, inflammation and all. And the most thing which is very significant about this silk fiber is that we do not have to use long screws for screwing them, since they have the sticking quality, adhesion quality. Therefore, usually we use 3 mm screws, but for 𝗓𝗏𝗏𝗏𝗏, if we use 1.5 mm screws, it is enough. So this is the benefit of using 𝗓𝗏𝗏𝗏𝗏. But gradually, we got to know that 3D braided structure of polylactic acid, this is even more beneficial, because if you see, this is not toxic. It does not have toxic substance at all. And this is a synthetic polymer. So it is, in short, we say it as PLLA. So this PLLA, it is non-toxic. It is very compatible with our body. That is, it does not inflame, and it bends. And even when we use 𝗓𝗏𝗏𝗏𝗏, it is tensile. This is comparatively more tensile. Let's see stem cells. These are really interesting. Stem cells are basically divided into two: embryonic stem cells and non-embryonic stem cells. Embryonic stem cells, they are what to say, pluripotent. That is, they can give rise to any types of cells in our body, but they are present at the initial stages of our life. But non-embryonic cells, they are multipotent. That is, they can give rise to a specific form of cells. And under this, we have adult stem cells. That is, mesenchymal stem cells. These are highly significant, because if you see, they are present in abundance in our body, in the adipose tissue, synovial tissue, bone marrow tissues. In all these, mesenchymal cells are produced, and this is a very good benefit for us. This is yet, up to now, we have not tried it on human beings, but it is tried on rats. That is, what we do, this is intra-articularly injected in the knee joint of the rat, and there was good improvement. That is, it got regenerated. The ligament got regenerated and it got joined, but yet we have to research about its strength. So it's not yet applied on human beings. Platelet-rich plasma. This is a very, what to say, this makes us think more. We use a silicon-made cotton-like structure. Usually we use cotton balls to wipe wounds, you see. Same like that, it is a silicon ball. This is sent inside our knee, exactly between the torn ligaments. Both the torn ligaments are tied with strings, and they are pulled such that the cotton ball is present between them, and it is sandwiched. Once it is sandwiched, blood from our own body, it's taken from our vein and injected into that silicon cotton ball-like structure. And once this is introduced, automatically the silicon ball is actually drenched with blood, and this causes our immune system to stimulate, and automatically it begins to clot. This gives us gives the ligaments the opportunity to develop itself and regenerate. This is applied up to now in rabbit, and it is not yet applied in human beings. It is found to be successful, and we are hoping that in the future this will really be useful to us. In conclusion, I would say that this reconstruction and regeneration, both of them have thrown light into sports persons, because this is found mostly in sports persons. It's like their career is doomed when their knee is destroyed. This gives them the opportunity to move higher in their lives. And actually, if you see, regeneration is more practical when considered to a reconstruction of ACL. If we see this in diabetic patients, their immune system is quite weak when compared to normal people. So if this causes pus formation and all in diabetic patients. So we prefer that we have to find new technology to overcome all these problems for diabetic patients. And moreover, this is not 100% successful, as I already said. Golden standards say that it's only 80% successful, and we find it only 40% to be successful. Therefore, we should find methods, and we should find a holistic approach to find a good cure for anterior cruciate ligament tear. Thank you, ma'am.
Thank you, ma. A nice presentation. So, for ligament treatment, normally use synthetic polymer. Whether this synthetic polymer has rejection or not, or it is 100% successful, because it is a synthetic polymer, no polylactic acid.
Ma'am, if you see synthetic polymer, obviously it is from outside our body, so there is rejection. But the least rejected is poly-L-lactic acid. 𝗕𝗶𝗿, it is rejective, but not so much. We usually select synthetic fibers which are quite compatible with our body.
Which method is best one? Which method is best one?
Ma'am, up to now, clinical method is human beings, ma'am.
Among three.
Among three, PRP is best, ma'am, but it is not yet applied in human beings.
What about stem cell? Method?
Stem cell,
it is 100% successful, no? Nowadays, doctors use to inject stem cells in ligament treatment.
Yes, ma'am, in ligament treatment, but when it comes to knee ligament treatment, you have synovial fluid which surrounds it. So growth of mesenchymal stem cells is quite affected, and yet we don't know the strength of the ligaments. So it is yet a question, ma'am. PRP, it regenerates
when we inject mesenchymal.
Yes.
When we inject mesenchymal cells in synovial fluid, the stem cell regenerate the ligament.
Yes, ma'am. No, ma'am. Yes,
so 100% is successful, is there?
No, ma'am. As far as I read in statistics, it says that PRP is more successful when we consider stem cell regeneration, ma'am. Because the synovial fluid, it affects the regeneration of stem cells.
Okay. Thank you.
Thank you, ma'am.
Reem Jalaluddin, one more question.
Yes, ma'am.
You said 3 options, that is silicon fiber, poly-L-lactic acid. Okay. So poly-L-lactic acid, how it heals the ligament tear?
And poly-L-lactic acid, it is actually the reconstruction. It is not regeneration. So what we do is we prepare a scaffold outside, and then as I said in clinical diagnosis, we drill a hole from the tibial end to the femur end, and then we insert it and screw them on both sides, ma'am. But the screw size is reduced. I said in 𝗕𝗶𝗿, we use 1.5 and when 1.5 mm screw. And when it comes to when it comes to PLLA, we use 0.5 mm of 0.5 mm size screw. So it is, I mean, the foreign product which we use, that is the screw, is reduced in size. Therefore, the inflammation is considered to be less. Therefore, we say that PLLA is more better when we consider to silk fibroin.
They use this treatment in humans, polyl lactic acid treatment.
Yes, ma'am. Yes, ma'am.
Is it successful?
Yes, ma'am. It is not 100% successful, but they say that it's up to 80% successful.
Oh, okay, ma. Good.
Thank you, ma'am.
Students, if you have any queries, please ask them. Okay, thank you, ma.
Thank you, ma'am.
Next, lot number 4, 𝗂𝗏𝗰𝗄 Malankara Catholic College, Kanyakumari.
Yes, ma'am. Good afternoon, ma'am.
Good afternoon, ma.
𝗂𝗏𝗰𝗄, you share your PowerPoint.
Yes, ma'am. Ma'am, it is not visible in the screen, ma'am. Okay, ma'am. My research topic is screening of antimicrobial and anti-cancer properties of actinomycetes isolated from vegetable degrading soil sample collected from 𝗕𝗏𝗰𝗲, Kanyakumari district. Actinomycetes are a group of microorganisms with the capacity of antimicrobial substance enzyme inhibitor. Antibiotics are commercially used as secondary metabolites produced by soil microorganisms. Actinomycetes are saprophytes. They are filamentous and sporulating. They produce branching mycelium. Most of the actinomycetes are source of antibiotics such as streptomycin. The soil samples were collected from 𝗕𝗏𝗰𝗲 market, Kanyakumari district. The isolation method was done by serial dilution agar plating method. Identification of actinomycetes were done by macroscopic, microscopic, and biochemical method. In microscopic method, I use gram staining method and spore staining method. In macroscopic method, aerial mycelium, submerged mycelium, color, and diffusible pigments were noted. The bioactive compound producing actinomycetes isolate were tested against 11 pathogenic bacteria and 8 fungal strains. Bacterial strains used were Staphylococcus, etc. The fungal strains used were Candida glabrata, etc. And the in vitro anti-proliferative effect of actinomycete species extract on cultured hepatic liver cancer cell line done by MTT assay. Next. In the result, after incubation, serially diluted agar-plated samples show different types of colonies. The colonies were streaked on actinomycetes isolation agar slant to maintain pure culture. antibiotic sensitivity with bacterial pathogens. Among the actinomycet isolates, the isolate A4 showed maximum activity against Vibrio vulnificus, followed by Bacillus megaterium. The inhibition zone diameter was 4 centimeter. The antibiotic sensitivity was with the fungal pathogens. The large inhibition zone was produced by actinomycet strain A4 against Aspergillus fumigatus and Aspergillus aculeatus. The inhibition zone diameter was 2.8 centimeter. Then the identification of the actinomycet isolate A4 was done, and the microscopic analysis were done. Morphological identification in this A2 and A4 were selected. In microscopic analysis, in gram staining, positive A2. A2 actinomycet isolate show purple, A4 show positive purple. In biochemical method, by the indole production test, methyl red test, Voges-Proskauer, citrate utilization test, etc. were done. And if A2 and A4 show positive and negative. In the anti-cancer assay, anti-proliferative effect of Streptomyces species on HCPG2 cancer cell line show. In this crude sample, 1 mule show 55.12% of viability, 5 mule show 53.12% viability, and 10 mule show 48.93% viability. Then the amplification of 16S rRNA gene was done, and the 16S rRNA gene show phenotypically identified as it is Streptomyces silicolor. In conclusion, thus this highly potent actinomycet is used in pharmaceutical industry for the production of valuable drugs. Thank you, ma'am.
Thank you, Ma.
Thank you,
Nice presentation. So,
Thank
what are the bioactive compounds in actinomycetes, Ma? What are the bioactive compounds present in actinomycetes?
It produces antibiotic capacity, ma'am. It's so many in it was I used many pathogens and bacterial pathogens and fungal pathogens. It showed inhibition shown against many bacteria and fungus, ma'am.
Okay. You represented inhibition zone as centimeter.
Yes.
Whether it is centimeter or millimeter. Whether it is centimeter or millimeter. Zone of inhibition.
Centimeter, ma'am.
Okay. How you identified viability of cancer cell?
By MTT assay, ma'am. By this anti-proliferative effect of Streptomyces by MTT assay.
By MTT assay or Tryphon blue exclusion method?
No, ma'am, MTT assay.
So we use MTT assay for identification of mitochondrial degeneration. So for viability test, which instrument is used for viability test?
Through MTT assay, cancer cell line, ma'am.
Cancer cell line. What is the basic principle of MTT assay?
MTT assay is the yellow dye which is reduced by cellular enzyme to blue product formosan. It is a colorimetric assay for cell metabolic activity.
You identify cell metabolic activity.
Yes, ma'am.
I am asking how you identified viability of cancer cell. Okay, ma.
Thank you, ma'am.
Bhageshri, one more question. You used 11 different bacterial and fungal pathogens. Yes?
Yes,
where you get these pathogens?
Through MTCC culture.
MTCC, okay. You confirmed the species 𝗕𝗶𝗿 in using 16S rRNA sequencing. Yes, you have sequence number. For the 16S?
Yes, ma'am. Sequence number.
But you didn't mention in your slide.
Oh, sorry, ma'am.
Okay. Okay, thank you, ma'am.
Thank you.
Next, lot number 5. 𝗕𝗂𝗂 Manonmaniam Sundaranar University, Tirunelveli. 𝗕𝗂𝗂.
We will share.
Ma'am, we have shared the presentation for you. Is it visible?
Visible, ma'am.
𝗕𝗂𝗂, are you here? Okay, you start your presentation.
Okay, ma'am. This is 𝗕𝗂𝗂. My presentation is on preliminary phytochemical screening and antibacterial activity of leaf extract of 𝗕𝗶𝗰 parviflorum, Tamil Nadu, India. First, I selected the plant. This 𝗕𝗶𝗰 parviflorum is of Rubiaceae family. It is commonly known as 𝗕𝗶𝗂𝗃 in Tamil Nadu. Based on the previous report, this plant material has many pharmacological importance. So I selected this plant. Then I went for authentication. Then I collected the plant sample and dried it. Then I went for 𝗧𝗿𝗄𝗃 extraction. These powdered samples were subjected for sequential extraction of secondary metabolites with a series of solvents. I used petroleum ether, ethyl acetate, chloroform, methanol, and aqueous extract. Then I checked for different types of the presence of secondary metabolites. Totally, I did 13 tests. In that, I observed the presence of different secondary metabolites in this plant. Then I went for antibacterial activity. For antibacterial activity, I selected 10 different pathogenic organisms, which was 5 gram-positive bacteria and 5 gram-negative bacteria. Gram-positive bacteria include Bacillus cereus, Staphylococcus aureus, Streptococcus mutans, 𝗕𝗶𝘁𝗲𝗏 and 𝗕𝗶𝗏𝗂𝗏 epidermis, gram-negative bacteria Pseudomonas aeruginosa, E. coli, Salmonella typhi, 𝗕𝗶cie 𝗁𝗂𝗏 marcianus. The sample was prepared. 10 mg of the sample extract was dissolved in respective solvents, and the sample was prepared as 100 mg, 200 mg, 300 mg, 400 mg by pipetting, 10 ml, 20 ml, 30 ml, 40 ml, and the final volume was made up to 50 ml by adding deionized water. The 𝗬𝗂𝗏𝗵𝗃 agar was prepared and autoclaved at 121 degrees Celsius. Then after that, approximately this 25 ml of the media was poured into the sterilized Petri plates and allowed it to solidify. After later 24 hours, the 10 different pathogenic cultures inoculum was spread to the plates. Then the 5 wells were made using the well borer, and the sample containing 100 mg, 200 mg, 300 mg, and 400 mg were loaded into the respective wells, and the control was used as a 50 ml deionized water, and it is incubated for 37 degrees Celsius for 24 hours. The minimum inhibition concentration of the petroleum ether extract against the pathogenous was observed in this. It showed mainly activity against gram-positive bacteria. In minimum in ethyl acetate extract against the pathogens, the minimum zone of incubation was observed as 20 mm. Then in chloroform extract, the minimum inhibition concentration was observed in 100 mg extract as 10 mm. In methanol extract against pathogens, the MIC was observed as 100 mg as 10 mm. And in water extract, it showed activity against gram-positive bacteria, and it showed activity in least 100 mg concentration. Thank you, ma'am. Hello. Thank you, ma'am.
Thank you, ma'am. Thank you for your nice presentation. I have 2 questions.
Okay, ma'am.
Which secondary metabolite is responsible for antimicrobial activity? You have identified, you have done phytochemical analysis, no?
Yes,
qualitative phytochemical analysis, alkaloids, flavonoids, terpenoids,
yes,
all are present in your extraction.
Yes, ma'am.
Which secondary metabolite is responsible for your antimicrobial activity?
Flavonoids will be responsible, ma'am.
Oh,
Flavonoids.
okay, ma'am. You have selected 4 solvents, I think.
5,
Aqueous,
ma'am.
5.
Aqueous,
among the 5, petiter, methanol,
petroleum ether, ethyl acetate, chloroform, methanol,
and aqueous. Okay.
Yes,
among this, which extract is the best one for antimicrobial activity, which shows maximum antimicrobial activity?
Methanol, ma'am. Methanol extract.
Methanol. Why methanol shows maximum antimicrobial activity?
Maximum zone of inhibition, but in water also got antimicrobial activity, ma'am.
Both aqueous and methanol extract is similar. It shows similar maximum antimicrobial activity.
Yes, ma'am.
Okay. So among these 2, which one you have to select for your research work, whether it is methanol or aqueous?
Aqueous, ma'am.
Why?
Because methanol, it is a solvent one. We have to buy and we have to use. Aqueous means it will be readily available for us to use, no, ma'am. So it is
okay, ma'am.
best.
Good.
Okay, ma'am. Thank you.
𝗕𝗏𝗁𝗂, why you specifically choose this medicinal plant? What's the major application?
Ma'am, basically it is good for a skin disease, ma'am.
Okay.
It has many, it has, based on previous report, it has many pharmacological importance, ma'am. So I choose this.
So what is the pharmacological importance?
Mainly in skin diseases, fever for constipation, leukorrhea, intestinal worms, diarrhea. For all this, these are used as indigenous, ma'am.
Okay, you select the pathogens, right?
Yes, ma'am.
This pathogens are skin pathogens, skin-related pathogens.
This is not purely skin related pathogens, ma'am.
Then why you select the pathogens?
First of all, I selected this path based on pathogenic, ma'am. Hereafter, I am going to do with the skin-related pathogenic organism, ma'am.
Oh, okay. Okay, ma'am. Thank you.
Okay, ma'am.
Next, lot number 6, Gayatri, Vivekananda College of Arts and Science, Trichandur. I repeat, Gayatri, Vivekananda College of Arts and Science. Okay, next, lot number 7, Suhaina Sanofer, Jamal Mohammed College.
Yes, ma'am.
Okay. Pass. Suhaina, you share your PowerPoint.
Okay, ma'am.
Gayatri, ma'am.
Ma'am, is my screen is visible, ma'am? Ma'am?
Yes, ma'am. Yes, yes.
Is my screen is visible, ma'am? Okay, ma'am. Okay. Good afternoon to one of our present here. I am Suhaina Sanofer. Now I'm going to present a presentation in the topic Critical Analysis of Pfizer-BioNTech COVID-19 Vaccine. So the general instruction about the vaccine is the name is BNT162B2 and it is manufactured by Pfizer and BioNTech company. So this is the mRNA type of vaccine. 2 shots will be given to the people and 21 days apart. And the shot will be given in the muscle of the upper arm. So here in short, how it works. First, the vaccine will train the immune system to recognize the disease-causing part of the virus. And the mRNA will be used as a vaccine, which is a genetic material. And this will give the immune system the time to design a powerful antibody so that those antibodies can neutralize the virus. So I'm going to explain now how the Pfizer and BioNTech vaccine works in brief. First, this is the mRNA. Here, the vaccine uses the messenger RNA, which is the genetic material. If this mRNA is directly injected into our body, it would be chopped by our natural enzymes present in our body. So to protect the vaccine, the Pfizer and BioNTech company wrap the mRNA into oily bubbles, which is made up of lipid nanoparticles. As you can see in this first picture, the mRNA is surrounded by the several layers, which is called lipid nanoparticles. And the second picture is the coronavirus, a piece of coronavirus which has a spike protein in it. And the first step is entering into the cell. So after the injection, the vaccine particle will bump into the cell and it will fuse with the cell. We can see in this picture that the vaccine is fused with the cell and it will release the mRNA. the cell molecule will read the sequence of the mRNA and it will start producing these spike proteins. And our mRNA from the vaccine will be destroyed by the cell. And the second step is spotting the intruder. Some of these spike proteins forms the spike and it will stick to the cells, and our vaccinated cell will also break some of the protein. Those protein will also stick to the cells. These protruding spikes and spike protein fragments will be recognized by our immune system. So when a vaccinated cell dies, the debris will collect all the spike proteins and protein fragments. It contains a spike protein and protein fragments. So those spike proteins and protein fragments will be taken up by an immune cell called antigen presenting cell. Here in this picture, we can see that antigen presenting cell is engulfing a spike protein. And our helper T cell will detect these fragments and it will alarm to fight against the infection. This is the second step. And the third step is making antibodies. And our helper T cell will activate the B cell. This B cell will attach to the surface of the vaccinated cell. I mean, bump into the coronavirus spikes on the surface of the vaccinated cell. So they will start to proliferate and they will pour out the antibodies. This is the third step. And the fourth step is stopping the virus. How it stops the virus from attaching to the other cells. The antibodies which is proliferated from the B cell, those antibodies will latch into the coronavirus and it will mark the destruction. It will block the spikes from attaching to the other cell. This is how it stops the virus from attaching to the other cell. And the final step is killing infected cell. So our antigen presenting cell also activate the another immune cell which is called as a killer T cell. This killer T cell will destroy the coronavirus infected cell. This is how this Pfizer and BioNTech vaccine works. And the sera of the people who had received this vaccine, they found that it neutralizes the COVID-19. And also this vaccine has been found to be very safe and effective in people those who have various kinds of diseases, and also for the children above 16 years, and can be also used for the people above 60 years old. The COVID-19 vaccine will protect us from getting COVID-19, but we all will be very scared to get vaccinated because we felt that we will have it will provide some side effects. Yeah, it's true, but there will be side effects in this vaccine also, but those side effects are not dangerous. They are the normal signs. They are the signs that is that our body is building protection against the infection. It is a natural protection called immunity. So these side effects are normal signs. So we need not to be worried about the side effects. And it's finally proven that a Pfizer BioNTech vaccine was 95% effective in preventing this infection. I would like to conclude by saying that billions of people today await a vaccine that is our best hope of avoiding this pandemic, said by Cyril Ramaphosa, the president of South Africa. We all are very scared because after viewing some misinformation in the social media, we are scared to vaccinate it. But COVID-19 vaccine will be very crucial in helping to end this pandemic soon and to return our lives to near normal. Thank you very much.
Hello. A nice presentation, ma. I have two questions to ask you. What are the antigen presenting cell present in our body?
Yes, ma'am.
Antigen presenting cell. What are the antigen presenting cell present in our body? This only present antigen presenting cell only present antigen to T helper cell, and T helper cell only provoke to activate B lymphocytes, isn't it?
Yes, ma'am. Yes, ma'am.
What are the two important antigen presenting cell present in our body?
B cell, ma'am.
B cell is different. B lymphocyte is different. And T lymphocyte is also different. So macrophages and dendritic cells, these two cells are very important to present antigen to T helper cell, and T helper cell provoke B lymphocytes. Okay. What are the side effects experienced by the public to take this type of vaccine?
The side effects like headache, tiredness, these are the mild side effects.
Only tiredness. They experience only tiredness.
Yes, ma'am. Some may experience like dizziness, but these are very common. Like this will increase our immunity, ma'am.
Okay, ma'am. Thank you.
Suhaina,
Yes, ma'am.
you said this is 100% successful, this vaccine?
No, ma'am, 95%, ma'am.
What about 5%?
Ma'am, the people, those who have an allergic reaction to the vaccine or any other medicine, this will not be suitable for them, ma'am.
Okay. Okay, thank you, ma.
Thank you.
Next, lot number 6, Gayatri, Vivekananda College of Arts and Science. I repeat, Gayatri, are you here?
Namaste, ma'am.
Namaste.
Hello. Is my voice audible, ma'am?
Yes, ma. Yes. Okay, you share your PowerPoint.
Hello. Hello.
Hello. We have shared the screen for you, ma'am. Is it visible?
Visible, ma.
Madam, am I audible?
It's audible, ma.
Yes, shall I start my screen sharing, ma'am?
Okay, ma.
Yes, ma'am. of this activity, 𝗓𝗏 flower extract. So why I have chosen this 𝗓𝗏 flower? In recent years, we can see fruits and vegetables have started achieving high attention in research field due to their therapeutic value, and it can also act as alternative for maybe existing antimicrobial agents. So due to this friendly and safe and eco-friendly nature, many bio-based agents, they are being used as antimicrobial agents, very significant. So 𝗓𝗏 plant, because banana, it is a large flowering plant in the genus 𝗓𝗏. Banana is also having many medicinal values, and the main scientific name for banana is 𝗓𝗏. So the why reason plant has medicinal and fundamental all part of plant useful human being, and also contains lot.
Gayatri, your voice is breaking.
Yes, ma'am. Is it okay? Am I audible, ma'am?
Your voice is breaking.
The of my current research work, it was like the presence of antibiotic antioxidant activity, and it also determines the phytochemical that are present in this 𝗓𝗏 flower. This is what my main aim and objective. And materials and methods what I have used in this 𝗓𝗏 banana flower, I want to carry on research. Even so many failures may come, we can able to easily buy it. So it's affordable. We can easily buy it from the local market. So that is why I have chosen this 𝗓𝗏. And second reason, even though it can be cheaply available, it is a lot of medicinal values. 𝗓𝗏 flower is having lot of medicinal values. So first and foremost, collection, sample collection. So since it is available from local market, we can able to buy it. Then the second step is screening of phytochemical compounds. This comes the major step in our research. So phytochemical screening of flower extract, it was done. So first of all, screening of alkaloids, it was done by Wagner's reagent method. And number two, screen of carbohydrate was done by using Mollis reagent. Screening of terpenoids was done. Then screening of quinones was done. Screening of sterols, screening of proteins was done by using a Millen's test. Then antioxidant assay, it was performed. And 𝗕𝗶PHF radical scavenging assay, it was also performed. And then in vitro screening. For in vitro screening, for bacterial culture, we have hired from Microtech Microbiology Laboratory from Coimbatore. So following bacteria we have isolated, we have got 𝗔. coli, Klebsiella pneumonia, 𝗕𝗏coccus aureus, Pseudomonas aeruginosa, Proteus species, then Salmonella species, and so many species were selected for using this culture. And immediately when we get all these isolates, it was inoculated into the chromogenic agar media, and it was observed for 24 hours. Then the next one, in vitro screening, first step, bacterial culture we have isolated, and second one, these cultures, it was subjected to well diffusion method. Then, so in well diffusion method, the containing plate, it was filled with one sterilized MHA acid, and then we allow it to solidify. Then we can able to enter each well was filled with a different concentration of extract, and the antibiotic ampicillin, it was taken as control. Finally, the antimicrobial expression, it was found in the restricted zone of incubated plates. Then I am coming to research and discussion. So the banana flower extract, which is prepared with various solvents using ethyl alcohol, butanol, acetone, and ethanol. So all these were used as solvent for preparation of banana flower extraction. And secondary metabolites, which was found in the plants, it helps to increase actually in the production of drugs, which can able to cure various diseases. That is the main reason why most of them we are taking plant in order to control this microbial activity or cancer activity, because plant is having so many secondary metabolites naturally present within them, which can able to cure various number of diseases. So if you see like saponins, tannins, Then flavonoids, alkaloids, terpenoids, phenols, all these are secondary metabolites which are found in plants naturally. So various methods we have used for identification of all these compounds like chromatography, especially high performance lipid chromatography was used in order to see all these compounds. Then phytochemical screening, why we are doing this phytochemical screening is considered one of the easiest and simplest method, but it also involves qualitative study. That's why the phytochemical screening is must in order to carry out any research. And based on this phytochemical screening, it was found that acetone crude extract of this 𝗔𝗂 flower contain more alkaloids or carbohydrates, flavonoids, saponins, phenols when compared to other three extractions. This acetone crude extract was found to contain much number of this alkaloids, I mean secondary metabolites. We can able see in the tablet column this alkaloids in a acetone extract, ethanol extract, butanol extract, ethyl acetate. We have used all these four extraction. Out of these four extraction, this acetone extract we can able to see so many number of alkaloids, carbohydrates present, flavonoids, phenols, saponins. Everything was given a positive result in this acetone extract. Why this banana leaf again? It was used because it's having a lot of pharmacological properties. The compound flavonoid in the extract is the main source, which is having so many medicinal uses, and it also protects us from various fungal infections like allergy, inflammatory disease, diabetics, and cancer. Another compound which we have found in this plant is saponin. The saponin is an excellent metabolite, or we can able to cure epilepsy or excessive salivation, fluorosis, and migraine.
Gayatri?
Migraine nowadays is... Yes, ma'am.
Kindly conclude your result, ma.
Yes, ma'am. Yes, ma'am. So 𝗕𝗶𝘁 assay was helpful for evaluating the antioxidant activity of various acetone extract. So these are a few results. We can able to say two types of solvents was used, acetone and ascorbic acid. Then antioxidant activity of banana flower extract. So these are various bacterial activities and its morphological characteristics.
Conclude your result.
So I just wanted to conclude like this banana flower I have used because plant is already considered a storehouse of natural antioxidant and can able to help in balancing the free radicals and antioxidant ratio in our body and prevent tissue damage. So this banana flower, why I have chosen, it is having a very cheap. We can able to do a number of experiments repeatedly. We can able to do, and also this banana flower extract can be used for various types of diseases like cancers, ulcers, diarrhea, Alzheimer's, etc. So my work suggests that banana flower extract can consider as a huge source of bioactive materials with extreme antibacterial and antioxidant activity. And thank you for all the participants over here, and thank you for the judges. Thank you. I just want to conclude with this. Thank you, everyone.
Thank you, ma'am, for your presentation.
Yes, ma'am.
Which constituents or secondary metabolite is responsible for antioxidant and antimicrobial activity?
Ma'am, once again, ma'am, here I am having very poor connectivity, and sorry if I have made you to worry this much.
You have identified so many phytoconstituents, secondary metabolites, alkaloids, flavonoids, terpenoids, all are present in your plant extract. I think acetone, it consists of most of the phytoconstituents are extracted in acetone compared to butanol, ethanol, and ethyl acetate, I think.
Yes, ma'am. Yes, ma'am.
Okay. Among these phytoconstituents,
yes, ma'am.
Which
Which phytoconstant you are asking, like which? Hello. Hello. Ma'am, actually, there is so many phytoconstants was secondary metabolites was alkaloids, carbohydrates, so many it was there. Among this alkaloids, carbohydrates, flavonoids, I think so flavonoids was shown a very good result, ma'am. Am I audible? Ma'am, is my answer audible?
Yes, yes. Why you didn't selected aqueous extract for your study?
What, ma'am? Aqueous extract.
Yes, yes, yes.
Ma'am, aqueous extract. If you
Okay, thank you, Gayatri. Next, lot number 8, Anita, Malankara Catholic College, Kanyakumari.
Good afternoon, ma'am.
Good afternoon. Please share your PowerPoint. Anita, share your PowerPoint and start your presentation.
Ma'am, sure, ma'am. Ma'am, not visible, ma'am.
Oh. Ma'am,
Ma'am, not visible in this PowerPoint. Ma'am. Ma'am, I am Anita, Research Scholar, Department of Biotechnology, Malankara Catholic College, Marigiri. Next slide. The topic is clinical phytochemical, antimicrobial, and physicochemical analysis of focus nucifera inflorescence. The next introduction. The coconut, the focus nucifera has been described as the tree of life or tree of heaven and nature's characteristic to man. Each part of the coconut tree can be used to produce items of value for the community. Focus nucifera is the dominant type tree belonging to the family Ericaceae. The common name of focus nucifera is coconut or coconut palm. Coconut is believed to have its origins to the Indo-Malaysian region from where it is spread throughout the tropics. Then next. In folk medicine, the first use of focus nucifera inflorescence is used in treating dyspepsia, diarrhea, dysentery, diabetes, etc. Indigenous people of tropical countries use young coconut juice is the treatment of stomach upset, diarrhea, and dysentery. The lauric acid content of focus endovas with the antimicrobial properties. It is useful in the treatment of digestive tract infections. Can be used in treatment of toothache and stomach. The present study mainly focuses on the screening of phytochemical and to check antimicrobial and physicochemical analysis. The next. materials and method, collection of plant materials. Then 𝗥𝗲 nucifera inflorescence were collected in the state of Tamil Nadu, India.
Anita, go to result and discussion. Go to result and discussion.
Ma'am, results were obtained by various analyses presented below. The phytochemical analysis of 𝗥𝗲 nucifera inflorescence using the spata, petroleum ether, chloroform and methanol. Phytochemical showed the presence of some secondary metabolites by the intensity of the colored solution and the precipitates formed on detection. The results were shown on the various extracts. The next slide, petroleum ether, chloroform and methanol. Then petroleum ether, chloroform and methanol, the various concentration. Then moderate concentration, various tests are there, steroids and terpenoids, and various tests of phenolic contents of tannins. These are moderate, low concentration and high concentration. Then next, then various tests. Antimicrobial activity. The screening of antimicrobial activity was screened above following organism: E. coli, 𝗥𝗲𝗰𝗲 pneumonia, Bacillus, Proteus vulgaris, 𝗕𝗏coccus, Pseudomonas. The methanol extract showed the highest antibacterial activity against Pseudomonas aeruginosa. Then next slide. Then methanol, chloroform and petroleum ether shows various organisms screened their antimicrobial properties. The methanol extract showed the Pseudomonas aeruginosa against 2.2 centimeter in their diameter of the antimicrobial activity, zone of inhibition. The next, physicochemical analysis. The various physicochemical analysis are there. Ash values, extractive values, and foreign matter. Then parameters and percentage of water soluble ash. Then 4.65 plus or minus 0.34. Acid soluble ash, insoluble ash 2.45 and 0.43. Then total ash 9.34. Then these are the ash values are mainly involved in this various properties. Then the result of 𝗥𝗲 nucifera inflorescence of petroleum ether soluble, alcohol solubles and water solubles. Petroleum ether solubles are 4.11 plus or minus 0.08. Alcohol soluble 12.23 plus or minus 1.35. Water soluble 21.95 plus or minus 1.54. Then next, determination of moisture content. The moisture content in the powder sample is inflorescence was found not more than 12%. Then moisture content is then important for the drug preparation. Then heavy metal analysis, the concentration of various metals in 𝗥𝗲 nucifera inflorescence. Then 𝗥𝗲 nucifera inflorescence, heavy metals include lead, mercury, arsenic, and cadmium. Then lead are 0.62 plus or minus 0.17. Then mercury 0.08 plus or minus 0.02. Arsenic 0.03 plus or minus 0.09. And 0.02 plus or minus 0.7. Then each analysis were triplicate in this standard deviation of these studies. And heavy metal analysis of various metals, lead, mercury, arsenic, and cadmium. In these are the next. Then concentration ppm per gram. Then discussion. The present study shows the presence of alkaloids and phenols and various concentrations of this study. Then petroleum ether shows the steroids and terpenoids are present in high concentration. The presence of steroids and terpenoids are moderate concentration in chloroform extract. The chloroform extract contains phenolics and tannins are present in moderate and low concentration. Then methanolic extract showed the steroids and saponins are present in low concentration. Phenolic and tannins are high concentration and moderate concentration, and carbohydrates are present in high concentration. Amino acids and proteins are present in low concentration. Then in a report, Diana and Kanjana 2012 reported that screening of phytochemicals showed the presence of alkaloids, flavonoids, phenols, and steroids. and tannins are present in Coccus nucifera flowers in aqueous spread. Then next step. The present study showed the ash content such as water soluble ash, insoluble ash, and
Anita, your voice is not audible. என்ன பண்ணலாம் க்ளோஸ் பண்ணிடுவோம். ஆமா மேம், நல்லா இருக்கு மேம். லீவ் ஆயிடுச்சு மேம், நெட்வொர்க் இதனால லீவ் ஆயிடுச்சு. ஓகே, நெக்ஸ்ட்
அவங்க லீவ் ஆயிட்டாங்களா?
ஆமா மேம், லீவ் ஆயிட்டாங்க. திரும்பி வராங்களான்னு பாப்போம் மேம். ஓகே மேம், நெக்ஸ்ட் போயிடலாமா? நெக்ஸ்ட்
இது வந்துட்டு ஒண்ணுமே பண்ண முடியாது, டச் பண்ண முடியாது.
Next, lot number 2, Reshma Rajan, St. Thomas College.
Yes, ma'am.
Reshma Rajan. இல்ல மேம், இது கிடையாது. Reshma,
audible? Yes, ma'am. It's audible.
Yes. Yes, you share your PowerPoint.
Ma'am, can you see this PowerPoint?
No, it's not visible.
Now it's okay, ma'am. Hello, ma'am.
Yes.
Ma'am, can you see the
visible?
Ma'am, I shared this PPT.
But it's not visible.
Now it's okay.
தெரியல, எனக்கு தான் தெரியல, யாருக்கும் தெரியல.
Ma'am, can you see the PPT?
No, no, ma. Okay, leave it. Next, I give the chance. Next, lot number 11, Asra Firdos, Jamal Mohammed College. Asra. Asra Firdos. Okay, lot number 12, 𝗤𝗏𝗏, Jamal Mohammed College.
Yes, ma'am.
Okay, you start your presentation. 𝗤𝗏𝗏, share your screen and start your presentation. It's already time up.
Okay. Ma'am, it's showing error, ma'am.
Can Shweta, can you share the PPT?
Your screen is showing.
அவங்க ஸ்கிரீன் தெரியுதா ரேஷ்மா? அப்போ
ரேஷ்மாவே கொடுங்க.
Ma'am, it's not my screen.
Reshma, are you ready?
Yes, ma'am.
Okay, start your presentation.
Thank you, ma'am. Respected teachers, good afternoon. I'm Reshma Rajan, research scholar, the postgraduate department of botany, St. Thomas College, Thrissur, Kerala. My topic is green synthesis of metallic nanoparticles using selected plants in Verbinaceae review report. As you know, the nanotechnology is a nano field. So the research that dealing with the production of plant-based nanoparticles. As we know, the nanoparticles are the great scientific interest due to the potential application in the biomedical, optical, and different field. The phytonanotechnology that based on the nanoparticles, the synthesis of nanoparticles from the plants, because of its eco-friendly production, especially eco-friendly production and it's the low cost. The experimental studies that prove that the importance of nanomaterials in growing research field, especially in the drug delivery, medicine, agriculture field, because of their environmental friendly nature, and it's easy to extract. It's easy to possible to extract under non-toxic chemicals. These are the some review literature or research work that are related to the production of phytonanoparticles from plant waste. The methodology that used in the extraction of materials, nanomaterials from plants are the collection of, yes, ma'am?
Go to your result.
From studies the phytonanoparticles, the synthesis of phytonanoparticles, the plant mediated synthesis silver nanoparticles increasing commercial demand due to the active microbes. The synthesis of nanoparticles from plant parts are many properties synthesized. Then the plant metabolites, the presence of plant metabolites and phytochemicals that imbue that can be used as a reduction metal ions, the formation of the clusters. Then the particles, the formation of nanoparticles, the phytocompound have been observed the present on the surface of nanoparticles. We can conclude that in the plant, the presence of secondary metabolites of a plant, they can be easily used as a catalyst or a reducing agent for the reduction of nanomaterials from the plant. Thank you, ma'am.
Question. Okay, thank you. Next, lot number 12, 𝗶𝗏𝗏.
Good evening, all. Myself, 𝗶𝗏𝗏 𝗂𝗏𝗏 from Jamal Muhammad College. Here I came to talk about biodegradable plastic from fish waste. First, plastics are everywhere and are used in almost all areas in modern day, including the making of cutleries, packaging, wrapping items, bottles, food containers, clothes, wearables, etc. and furniture, among many other items. Their wide usage is because of their versatility and 3D modeling, durability, and ease of work within various manufacturing and production process. In short, it is really difficult to imagine our daily lives without plastic. Okay, how are biodegradable plastics made? Biodegradable plastics are made in a way that they can break down or degrade when exposed to the sun, ultraviolet radiation, enzymes, bacteria, water, or wind absorption. They are made from renewable raw materials or all natural plants or animal materials such as orange peels, corn oil, soybean, microorganism, or starch. The industrial processing of biodegradable plastic is similar to the manufacturing of ordinary plastics, only that material used differ, and for biodegradable plastics, they are the material that can easily break down and decompose. Various advantages of using biodegradable plastic. First one, biodegradable plastic are easy to recycle. These type of plastics not only take less time to decompose when they discarded, but can only be easily recycled through an organic process. They are also non-toxic since they have no chemicals or toxins. Recycling helps to less landfill problems, and besides, the recycled bio-waste can be used as compost or a renewable energy for biogas. Uses of biodegradable plastics. Biodegradable plastics are used in many areas of our daily lives today. Some of these uses are, they make shopping bags, compostable waste collection bags and trays, nets for fruits, meat, and vegetable, and the medical department used bioplastic to make screws, pins, and plates materials for pills and capsules. They are used to make disposable catering services wares. They also make items such as bottles, tea bags, jars, air pillows, pens, pencil sharpener, mulch films. From these wastes, I have chosen fish waste. A reason to choose fish waste. India is one of the global leaders in fish production.
Lila, you are not audible, Lila. Lila, are you there? இப்போ கேக்குது. A reason to choose fish waste.
Okay. Yeah. Reason to choose fish waste. India is one of the global leaders in fish production, where fish export reached 11.5 lakhs in 2015 to 2016, of which roughly 50%, that is 5.5 lakh tons, has been disposed as waste. Fishing generates large quantity of waste daily in fish processing industries. Also, fish leather is a part of an ancient tradition, especially in East Asia. The advantage of this material is that it is the most source of unusual leather. But production process can use of fish from food industry. The organic fish waste is normally headed for the landfill or incineration.
Go to your conclusion.
Yep. Okay, ma'am. I have used red algae to bind.
Nila, you're not audible.
Thank you, ma'am. Can you hear me, ma'am? Hello, ma'am, can you hear me?
Okay, ma. Thank you.
Thank you.
உங்களை கோஹோஸ்ட் ஆக்க சொல்லி இன்னும் மேம் ஆக்கவே இல்லை. ராகுல் காந்தி, அம்சா பர்வீன் இருக்கா, அவங்களும் கோஹோஸ்ட். அம்சா பர்வீன் நடத்திட்டு. நெக்ஸ்ட் அம்சா பர்வீன், ஜமால் முகமது காலேஜ். அம்சா பர்வீன், are you ready?
மாஃபியா ரெண்டு பேரும் கோஹோஸ்டா மாத்திருக்கோம். நான் பிரசன்ட் பண்ண வருவாங்க. சுதா அண்ட்
ராகுல் காந்தி. அப்பே
பாண்டிச்சேரி யூனிவர்சிட்டி.
ராகுல் காந்தி கொடுக்கல. Ms. Sudha, are you ready for presentation?
Yes, ma'am. I'm ready, ma'am.
Okay, share your PowerPoint.
Is it visible, ma'am?
Visible, ma.
Is it visible, ma'am?
Visible, ma. Hello.
Good afternoon, dignitaries. Yes, ma'am. Good afternoon, dignitaries.
Good afternoon.
Good afternoon, ma'am. Good afternoon, ma'am.
Good afternoon, ma.
I am Sudha, Department of Biotechnology from 𝗕𝗏𝗏 College, Trichy. Yes, ma'am. Yes. My topic is evaluation of phytochemical screening and antimicrobial activity of methanol extract of 𝗖𝗏𝗏𝗿𝗏. Introduction. We have selected for specifically in Alzheimer's disease referred to chronic neurogenetic diseases that usually starts so gradually affected over a time in aged persons. It usually affects not only the aged person and middle-aged persons also. This deterioration of brain cells even leads to service mental deterioration, dementia, and death. It causes a combination of genetic life cycle and environmental factors that affect brain over time. Problems with the brain proteins that fail to function normally, they disrupt the work of brain cells and unleash.
Ma'am, can you unmute, ma'am?
Shall I continue, ma'am?
Yes, ma'am. Ma'am,
Yes,
share
ma'am.
your screen, ma'am.
Okay, ma'am. 1 minute, ma'am. I have shared, ma'am. Is it visible, ma'am?
No, ma'am.
It's not visible, ma'am.
Time match. Just conclusion சொல்ல சொல்லிருவோமா மேம்? சொல்ல சொல்லிருவோமா?
Hello, ma'am. Is it visible, ma'am? Ma'am?
Ma'am, it's not visible. Okay, you just conclude your result, ma'am.
Okay, ma'am. Sure. So I have got from the phytochemical analysis some of the positive results from carbohydrates, saponins, flavonoids, quinones, and steroids. From the GCMS report, I have got a very good unsaturated fatty acids containing linoleic acid. At the time of pandemic, I have reworked for the I have entertained the same sample, my gram antibacterial as well as UV visible analysis also. It's a part of my work. The same 𝗖𝗏𝗏𝗿𝗏 doses I have given to the model organism is known as a very good, easily available zebrafish. I have got to for acute toxicity analysis. I have decided for 4 different groups. First one is control normal feed. Group 1 contains 100 mg per kg, like that. Group 2, 150 mg per kg. Group 3, 200 mg per kg. Have given the doses for each groups. Then I have go for a liver function test. After 14 days of incubation, I have collected brain, gill, and liver. I have analysis of histoarchitecture studies also, and got good results for histoarchitecture in brain cells and gill, and then liver functions also. This is the overall my views about Nigella sativa antioxidant activity. It has a highly potential bioactive compounds. It's safe to develop as a lead active compound. Thank you very much, ma'am. Hello, ma'am.
Hello, ma.
Yes, ma'am.
I have two questions.
Yes,
How
ma'am.
you fixed your dosage? 100 milligram per kg body weight and 150, 200 and 250, I think. You have
Yes,
fixed
ma'am.
4
Yes, ma'am.
dosages, no?
100. Yes, ma'am. I have referred more journals for the previous work. They have given 75, 150, like that, ma'am.
Okay.
I have slightly changed 100, 150, and 200, ma'am.
200. So
Yes, ma'am.
you have not done acute toxicity study.
No, ma'am. No, ma'am. I will decide to continue the same for acute toxicity also, ma'am.
Okay. How you treated control? Control.
It's a normal feed only, ma'am.
Okay. Okay.
Oh, yes, ma'am.
You have done
Yes, ma'am.
antimicrobial activity also.
Yes, ma'am.
Antimicrobial activity and LFT, liver function test.
Yes,
You have
ma'am.
done
Yes,
liver
ma'am.
function test. What are the parameters you
Yes,
have
ma'am.
selected? What are the parameters you have selected for liver function test?
For our human being parameters only, ma'am.
What are they?
Like SGOT, SGPT, serum alkaline phosphate, like gamma GT, like that, ma'am.
Okay, ma. What is the standard drug used for this assay? Standard drug.
Donofacil, ma'am. For a liver function test, ma'am.
For a liver function test.
For liver function test, SGOT serum we have to given to biochemical laboratory, ma'am.
Okay, ma. Okay, ma.
Yes,
So commonly for liver function test we use silimarin as a standard drug.
Okay,
Whenever you do any pharmacological activity, you must use standard drug.
Okay, ma'am.
You compare
okay, ma'am. Sure, ma'am. Yes, ma'am.
your result with standard drug as well as control. Okay.
Okay, ma'am. Yes, ma'am. Yes,
Okay,
ma'am.
ma'am.
Thank
Okay,
you for
okay.
your
Thank you.
suggestion,
Thank you.
ma'am.
Thank you.
Thank you, ma'am. Thank you.
Thank
Ma'am. Yes, ma'am. Thank you. Yes, ma'am. Yes, ma'am.
யாருமா இவங்க? சுதா.
கன்க்ளூட் பண்ணி.
Thank you, ma'am, and thank you all the participants. Thank you, ma'am, for being the judges and evaluating with utmost care and sincerity. Now we move on to the next session. முடிச்சிட்டேன்ல இப்பதான் முடிச்சேன் சொன்ன 3:30 ஆயிடும் 3:30 மேலேயே ஆயிடுச்சு Let's move on to the next session.
Now we have the third invited lecture. Scientists have become the bearers of the torch of discovery in our quest for knowledge. Now I kindly call upon Dr. 𝗓𝗂𝗏𝗏 ma'am, Assistant Professor of our department, to introduce the invited speaker.
Good afternoon, everyone. I feel extremely honored to introduce the keynote speaker of this session, Dr. 𝗓𝗏𝗏𝗏𝗏. As one of the most distinguished scientists, Sir completed his undergraduation and post-graduation from CCS University, Meerut, Uttar Pradesh, and completed his Doctor of Philosophy from Wildlife Institute of India, Dehradun, and Kurukshetra University, Haryana. He is having 5 years of research experience and 8 years of professional experience in the institutions like Wildlife Institute of India, Dehradun, National Daily Research Institute, Karnal, National Bureau of Animal Genetic Resources, Karnal, Amity University, Noida, IARI, New Delhi, Institute of Animal Science of CAS, Beijing, Chinese Academy of Sciences, Kunming, and University of Padova, Italy. Sir also received prestigious awards like DST Young Scientist in 2013, Fellow Chinese Academy of Sciences in 2016, Inspire Faculty Award in 2017, INSA Young Scientist Medal in 2018, and he is also a certified wildlife forensic scientist. At present, he is working as a scientist C in Center for DNA Taxonomy, Molecular Systematic Division, Zoology Survey of India, Kolkata, and supervising 6 PhD candidates, and also completed 2 major research projects funded from Chinese Academy of Sciences and Department of Science and Technology. Presently, Sir is handling 4 major research projects funded by US-based NGO Conservation Force, Department of Science and Technology under Inspire Faculty Scheme and Zoology Survey of India. Apart from this, he is also acting as a co-investigator for many funded projects. His field of research interests are conservation of landscape genetics, genomics, metagenomics, ex situ conservation, assessment and restoration of biodiversity, especially threatened mammals in eastern Himalayas. And he significantly contributed to bring Kashmir red deer and upgraded the conservation status from least concern to critically endangered under the red list of IUCN. He has published more than 60 research articles in reputed national and international journals like PLAS ONE, Cell Research, Scientific Reports, with overall impact factor of 80.04 and Clarivate in Analytics impact factor of 137.206. And few of his contributions also recognized by DSG SSC Group. Apart from this, Sir also presented several papers in international conference and symposium. He is an academic editor in PLAS ONE journal, member in editorial board in Forensic Science International, Animals and Environment journals, and also member of review committee in several journals like Scientific Reports, PLAS ONE, Mammalia, Conservation Genetic Resources, Current Science, Mitochondrial DNA Part A and B, and Food Control. In addition to this, he is also a member of Society for Wildlife Forensic Science, IUCN SSC Deer Specialist Group, and a lifetime member of the International Society of Animal Genetics, International Society of Zoological Sciences, and Society for Conservation of Domestic Animal Biodiversity. With this brief introduction, now I invite Dr. 𝗓𝗏𝗏𝗏𝗏 to take over the session. Please, sir.
Yeah, thank you so much, madam, for a detailed, elaborated introductions. I really, after a long, I heard something about that elaborated introduction of mine. So it is always, feel great when you listen actually such a big introductions. But still, I believe I'm still a learner, and yeah, we have been doing several projects in ZSI, and the number of students, they are working with me. And as you have well elaborated my introduction, I started my career from Wildlife Institute of India. And actually, before going into this, I just want to inform all the students here that basically my formal education was in biotechnology. I had my BSc biotechnology, MSc in biotechnology, and my PhD was also in biotechnology. But now I'm fully, devoted and dedicated to doing wildlife conservation and conservation biology. So though these two areas, seems pretty different or sounds you very, different. But I tell you, how we can use the molecular biology tools in the species conservation. When somebody talk to you about the wildlife, you always think of, going to a national park, protected area, and undergoing field survey, and watching the animals in the field, recording their behavior, camera trapping, so on. So most, most of the us who was not exposed to the wildlife, they don't understand how we can use the molecular biology tool in species conservation. So that way, this my topic would be a little different, and I hope that this would be very interesting also. And I have used very good photographs also to keep you connected with the wildlife and biodiversity conservation. And at the same time, I will also touch upon all the molecular genetics tools. So that is why my title is also sound like role of conservation genetics in wildlife conservation. So I know all of you must be tired because it's a long day session and you are attending this since morning. So before starting my formal talk, I would like to thank and acknowledge Dr. 𝗕𝗫𝗁𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 Haji Dr. 𝗓𝗁𝗁 Principal, 𝗬𝗓𝗁 College. Actually, it is a great initiative, and I'm really, feel very glad to read the invitation when it says second international conference. So that means the college has already, taken initiative sometime back in year, and they have already have this kind of a conference. So I believe if this is how the college is progressing, performing, because I was attending all the sessions since morning, and I was really very impressed. I mean, this is how the faculty should work in the college, so that the young generations here, all the students, master students, they get benefited, and not only to the they have confined to the 𝗬𝗓𝗁 College, but they have also incorporated students, involved students from other colleges in Tamil Nadu or elsewhere, I believe. So this is actually a good interaction among the faculties, students, and also an opportunity for me particularly to interact with you all, and to showcase my work, and understand what you are doing. So I was attending all the small speed talks made by the students, and many were on the antimicrobial or antibacterial activity of different plant system. And I really like that ligament tissue engineering presentation. It was little out of the routine done. So though it was kind of a review paper only, but yeah, I really took interest in this, and then Pfizer vaccine, how it is impacting. So fine, I think I would have enough time because the last presentation. If not, I request the organizers to kindly excuse me, because I would be talking a little more, and this is the last presentation, and I am listening everybody who has come before me. So now whether you listen or don't listen, but you have to give me enough time to speak about my topic. So with this, I would like to share my screen. I hope it is well audible and visible to you. I'm now sharing my screen. I'll appreciate if somebody just confirm whether my slides are visible to you.
Yes, sir. Everything is clear, sir. Everything
okay.
Is clear. And go ahead.
The beautiful animals, right? On this, if you see on my extreme left, this is red panda, is the pangolin, the ibex, and the Arunachal macaque is one of the charismatic and the endemic species of macaque found in the eastern Himalayas. So I
Sir,
work on
sir,
hello.
Still, we are not getting any screen.
You're not getting my screen?
No, yes. You are visual, but screens are not visual. PowerPoint is not.
Okay, let me share it again.
Yeah.
Is it
now
visible now?
Yes, of course.
Is visible too now?
Yes.
Okay. So here are some beautiful pictures here. There's a different species found in Himalaya. So is a red panda. And if you see the today's Hindu newspaper, we are on the front page. Yesterday only one big story that we have published, and the story is covered in the Hindu. So we are on the front page on the today's Hindu newspaper. Is the pangolin, the anteater. The animal is very different. It is made up of the keratinized scales, the entire body covered with the scales, the Himalayan ibex. So why I'm showing you all this picture, just to bring you interest. I mean, when people think the biotechnologies is not only confined to the lab, and it's not only confined to jumping behind the genes and the enzymes, but there is a other beautiful world also where you can punch upon and you can really do marvelous job in species conservation using biotechnological tools. So I'm one of the example of this, what I'm using the modern tool in species conservation. So before discussing this, I would like to brief you about the wealth of the institute that we have here. So Geological Survey of India, as you all know, it is a part of the Ministry of Environment, Forest, Climate Change. And then we have a 16 regional center with a headquarter in the Kolkata. So 𝗕𝗍𝗍 is known for the sample collections, the specimen repository that we have, and the National Biodiversity Authority, they have authenticated 𝗕𝗍𝗍 as a national repository. So we are the country repository for the all faunal specimens. In 𝗕𝗍𝗍 itself, we have about 17,000 type specimens. So when I say the type specimen are those on which the species are named. So we are fortunate and lucky enough to have this much of 17,000 type specimen, and approximately 5.5 million specimen, including 11,000 slides that we have. And these are all the wet and the dry collection that we have in 𝗕𝗍𝗍. Yes. Is it all okay? Is good to go?
Yes, sorry, you can go, sir. Sorry.
Okay. And then because 𝗕𝗍𝗍 was first established by the Britishers in the colonial period, so we have specimens from more than 56 different countries. So institute was established in 1916, and this is a type of DNA barcode that institute has generated. So we have the little old data. We have now generated over 6,000 DNA barcodes in the country, and they comprise of different taxa and different groups. 𝗓𝗏𝗏𝗏, like all the pests of the animal kingdom is categorized in 𝗓𝗏𝗏𝗏, and then lepidoptera, all the butterflies and moths. So we have a different type of taxa and group for which we have already generated these many barcodes. So it is kind of a digital sequence information, and DSI, we say, it is used in various aspects. Like DNA barcoding, we have already completed the DNA barcoding of the scheduled animal, that is in the Wildlife Protection Act of the country. And then we did DNA barcoding for the endemic species and for the wildlife forensics. So we have already generated a species signature. When we say DNA barcode, it is basically the species signature. So we have generated species signature for these many species. Now I'm going to brief you about to some of the projects where I'm involved, right? And I would be then discussing some of the key findings of these projects. Okay, so these are the 3 projects listed. The first project is the conservation of threatened vertebrates. So we are working in the entire Indian Himalayan region, and then we are collecting samples. I mean, we cannot, catch the animal. So what we are working with the indirect samples. We are collecting fecal sample of the animals, and then we are doing all DNA analysis using the DNA extracted from the fecal matter, whether it is a carnivore, herbivore, or any, birds also. So the second project I am involved with, this project is funded to me under DSC Inspire Faculty Award, is the conservation of red panda. So we are working in Eastern Himalayas, a very beautiful, the panda species, the true red panda, true panda. While the giant panda, which is in China, is not basically a panda. It belongs to the 𝗕𝗏𝗏 family, where bear are listed. So we have a second project under me is on conservation of red panda, and then genetic monitoring of Asiatic black bear. So these are the three projects where I am involved, and I would be touching up the key findings of these three projects. So I'm involved in developing the spatial database of threatened vertebrates, because until unless you don't know the distribution or the presence of the threatened vertebrates in different systems, it would be difficult to have a management plan for the entire protected area or a bigger landscape. So we are monitoring the free-ranging wildlife. So all these free-ranging wildlife means the animal which freely move in the wild. It could be the tiger, leopard, bear, snow leopard, anything, I mean, in the forest. So we do sign surveys, we do camera trapping. So camera trapping is kind of a system where we put cameras and leave them into the field and they are automatically triggered cameras using RFID technology. So whenever animal passes, the camera trap take the pictures and we identify whether what are the presence of the animal or species diversity in those areas. We do DNA analysis using the scat sample, and I would be much focusing on the DNA analysis. And then we are setting up the long-term monitoring protocol. So if we have now we have a baseline data, so we can predict, I mean, after a gap of say 5 years or 10 years, that can be monitored. So we have already set up some permanent monitoring plot in Himalayan region, and these can be monitored as a long-term basis, and we can see how the genetic diversity is affecting over the different time scale. And then we are also working with the forest department to enhance the capacity building, and working with the wildlife department, and also get involved with the local institutions, NGOs, for the implementation of the different activities of the projects. And then also working on the socio-economics aspect, recording the ethnological knowledges, practices that always retained by the locals. This is the key site in Himalayas where we are working upon. So is the 𝗓𝗏𝗏 is a 6 study site in 5 districts. So one study site in 𝗓𝗏𝗏 is in Trans-Himalayan region. So 𝗓𝗏 falls in the Trans-Himalayan region. 𝗓𝗏 is in Greater Himalayas. 𝗏𝗏𝗏𝗏 again in Greater Himalaya, and East Darjeeling, West Kameng, and the East Siam. These are the sites where we are working on. And so if you know the biogeographic classification, I'm not going into detail. So these fall in a different biogeographic zone of the country. And this is sort of the, adaptive framework for undertaking the species monitoring. So first we classify the habitat, different habitat, using the GIS-based tools. So before planning for any field study, we sit in the GIS laboratory. We digitize, we open the maps, the landscape map, and we plan our study, which areas we need to visit based on the accessibility, terrain, climate, different seasons, and considering the activity pattern of the animals. So once we have the map, then we stratify our sampling, that these are the areas where we need to visit, where we have to concentrate our efforts. Then we visit those areas, we collect secondary information about the presence of the animal, we do PRA, remotely sense data we assess, and then we do intensive survey and the extensive survey. So extensive survey are the single time efforts. We just go visit and go for the science surveys. When I say science survey, that means we are walking into the tree lines or maybe the prominent paths in the forest, trails sometime, and we collect the presence of the animal. So we collect the signs, maybe the foot marks, maybe the scratch marks, and the the fecal material. So those things we collect, and this data we use for the genetic profiling from the fecal samples. And once we find those evidence, the presence of same area, then we do an intensive visit, is a repeated sampling in those areas to then understand the population size and abundance, species diversity. So first we go for the extensive survey based on the information collected about the species presence, and then we then do repeated sampling in different seasons to understand the activity pattern and also collect sample for genetic profiling. So then we determine the species. So this is again the same, the pictorial presentations. We go and map the areas. For example, for the red panda, we map the bamboo cover where the bamboo is distributed. Then we visit those areas, collect samples, we do the genetic analysis, generate digital sequence information. We also work with the forest department and law enforcement agency. We make aware them, and we share our SOPs, the standard operating procedures for dealing the crime scenes. And then, because I am also involved in institute and the coordinator of wildlife forensic activity, so we do the crime scene investigation and forensic analysis, also do DNA profiling, DNA fingerprinting, and we keep organizing, raising workshops and awareness program in different forums. So this is how we plan the study. So we is a kind of a map on the Eastern Himalayas. So we define the areas we need to walk on, and then we collect the fecal sample. This is a fecal sample of red panda. So from the fecal sample, we extract DNA, and from DNA we put PCRs with the specific genes and markers. Once PCR we do, then we do sequencing, and based on the sequencing we do different analysis, population genetic analysis, to understand how the animals are moving in the landscape, what is the level of genetic diversity, what is the level of population assignment, and I mean how they are distributed and using the landscape. So we do the landscape genetics approach also, where we use the GIS tool and the DNA based data, and then we mingle these two information and define how the animals are moving in the landscape. It's called the landscape genetics. So far, I mean, it's a 3-year efforts. These many camera traps, about 12,700 trap nights we have already worked upon, and so this is the effort that we have done so far. So I'm talking about the DNA analysis. In last 3 years, we have collected over 5,000 fecal sample, and those samples have been processed in lab for the DNA profiling. So it's kind of a field activities that the student has done. So it's kind of a getting some feces. So the student is collecting the fecal samples, recording the data. Here they are, capturing the fishes with the nets, and then here they are collecting the habitat information, putting the camera traps here for the photographic evidence of the capturing the photographic evidence. And it's from the 𝗖𝗲 region where we do not have any vegetation. So this is how we are putting the camera traps. This is sort of a devices which actually capture the images of the animals. Whenever any animal passes through this, the cameras capture the animal picture. So these are the feces or some evidences. So some ungulates has been visited these areas. So ungul droppings is the red panda droppings. Some canid dropping, and this is a pug mark of a leopard, I believe. So these are all the sign and feces. So when we do the sign survey, we record all this information about the species presence. And this is how the camera trap, as I mentioned, these cameras, take the photograph of animal as it passes through. So we have placed cameras in different elevation zone. And you see we have a camera trap from approximately 500 meter elevations up to the 5,000 meter elevation in different state and the district. So I believe that this is the highest elevation that we have put camera trap in this our exercise. And this was actually in the 𝗖𝗲 region, and then we have captured several species. So this is a snapshot information about the 5 dominant species that has been captured using the camera traps. So the barking deer, 𝗂𝗲𝗏. So I'm not going into this. It's not very much related to the biotechnological students. But this is some pictures of camera trap photographs where we have recorded the musk deer, the brown bear, and the fox, red fox, and snow leopard, and the barking deer, mako. So we have identified several species for the detailed population genetic analysis. I will take them one by one. So it's for the red panda. So if you see the red panda, this is a red panda distribution. So it's found in the central Himalaya, is in the North Bengal and the Sikkim, and is the Arunachal Pradesh. So this is the 3 states where the red panda is distributed. So, and the selected altitudinal range where we have put maximum efforts was the 2,400 meter to 3,200 meter. So these areas, which is in the category 2, this has been surveyed upon not all grids has been surveyed. The grid size was about 5 kilometers. So this we planned before planning any, stratified the map before planning for any field studies. So we based on the logistically possible and those terrain, we have focused more upon in this second type of altitudinal range. And then we collected this SCAT. This is how we have collected these SCATs, right? And these SCATs were sun-dried and then stored in the silica gel. Silica gel work as a very good desiccator, so it absorb the water content, moisture content, and the SCATs are safe for during transportation. So we have conducted 5 field survey for red panda fecal collection. So this is the site where we have collected about 115 SCAT samples. And then we did the DNA analysis. So there are specific protocols for undertaking DNA analysis using the fecal DNA, fecal matter. So there are certain commercialized kits are available. So we extracted DNA and then did the DNA profiling with the microsatellite markers. Here you say we have tested about 48 STRs, is also known as short tandem repeats. So out of which we have identified 24 individuals using 9 STRs. So this is basically the data of the 9 identified red pandas in the Eastern Himalayas. So this is kind of a individual identification exercise. So it's little technical. I'm not going in detail how we do this, but out of 48 STRs we chose, we use 9 STR for the individual identification from this data. And then we have also done a landscape genetic analysis. So it is kind of a modeling and mapping the habitat. The red area is the prime suitable habitat of red panda in Kanchenjunga landscape. This is basically the Kanchenjunga landscape part. Is the upper part is in the Sikkim, and then is about the north West Bengal. So we have modeled the suitable habitat of red panda in this region, and on the same region we also collected samples, red panda sample, and did the DNA analysis. So here you see the different currents. This current shows basically the amount of gene flow from one location to another location. So if you see this red or yellowish red is become the high gene flow. So if you see there are 2 distinct populations here. This is basically a Singalila National Park, and here is the Nira Valley National Park. So I just wanted to say these 2 populations are separated out. They are isolated. They are disjunct population. And if we do not have this genetic analysis, we won't be able to say this. But since we have done a gene flow analysis, and this current flows shows the gene flow, they coincide with the gene flow among the populations. So you can say this is one good population lies here, another good population lies here, and they are loosely connected. So now if I have to give a management plan or a recommendations to the manager how we can have a good population of red panda in Kanchenjunga landscape, I would say here is the region where somebody has to do plantation or has to add upon the red panda suitable habitat. And red panda is largely bamboo feeder and is an arboreal species. So I would suggest the administrator, the manager, they have to work upon these areas. They have to plant the suitable habitat of red panda so that it will support the movement or the physical movement of the red panda, so that this would be a contiguous population. If the population get disconnected, then there would be chances of inbreeding. The population become even super isolated, and there would be chance of increase inbreeding and the local extinction. So this is how the same thing has been predicted. So we found as good gene flow. So there are meta population exists in this landscape. I hope you would be able to follow it with me. And when I say the structure, so structure analysis talk about how many different populations are there. So one color shows one population. So I can see there are at least 4 color are here. So this is basically assignment of the population. So in this landscape, wherever we have collected samples, we found at least there are 4 populations. So it's a population genetic assignment. So we got at least 4 genetic cluster in this landscape. So is like to understand the directional analysis, whether the directional of gene flow analysis. So whether the gene flow from either region, from west to east or east to west, is random, or is there any directional of gene flow. So this, because many of you would not be familiar with the landscape, so I just say this is sort of a gene flow analysis, and we try to understand the directional of gene flow in this landscape. So this is, I request if you can read the today's Hindu newspaper, and even not if you don't get a physical copy, search on the Google, you will find this on the newspaper itself. This study was published in Mammalian Biology, and this is a journal published from the German Society of Mammal Biology. So we have proven that there are two species of red panda. It's called Himalayan red panda and the Chinese red panda. Himalayan red panda is found up to the Xiang River. So we have proven this. If you follow my cursor, this is a Xiang River. So in the east side of the Xiang River, this is a Chinese red panda. It's called 𝗕𝗏𝗏𝗏. And below the Xiang River, in the western part of the Xiang, all this up to the Nepal is a Himalayan red panda. This is a different species, 𝗕𝗏𝗏𝗏. So we have proven this through the genetic analysis. This is basically a phylogenetic analysis. So we have sampled all these areas. So this is the red panda that has been sampled in China. So because here last here is the Arunachal Pradesh, then comes the Yunnan and the Sichuan province of China. So we have proven this red 4 dots fall in the in India is in the Dibang district of Arunachal Pradesh. So here we have a Chinese red panda. So we have established this. India is home for both the species, and this is all using the molecular biology tools. We have done a phylogenetic analysis using the mitochondrial markers. So earlier thought this is the Nuqiang River, which fall in Yunnan province. So Nuqiang River is the barrier, and we do not have a Himalayan red panda below. Chinese red panda below this Nuqiang River, but we have. This month only we got this paper published. So and one article published in Scientific Report just on this, where we have talked about the Pleistocene glaciation, how it is affected the demographic pattern of the red panda. This is another species we are working on. It's the Arunachal macaque, and this is a very recently described macaque species from the eastern Himalayas. And this was probably described in 2005, around 2005, and was only reported from 2 districts in Arunachal Pradesh, the Tawang and the West Kameng. So our team has went these districts and collected samples. So this triangle is the sample locality where we got the samples. We have again done the scat analysis. Scat is called the fecal material of any carnivores. So because macaque is like a omnivore in nature, so we have used the term scat. So we have done the sample collection, and then we found 11 individuals based on the 15 STRs data. So we have also done a STR analysis here. So this is a sort of STRs, and we found there is a good gene flow between these two populations. So it's not isolated like the red panda, but it's a good gene flow between the two populations. It's the first time that we have done genetic assessment of the Macaca munjala and Arunachal macaque in Arunachal Pradesh. So now is there another fascinating results. You must be attracted to understanding this data. So we thought to also understand the gut microbiota. So gut microbiome is like something which exists in our alimentary canal, in our stomach. So there can be symbiont microbes also, there can be some parasitic also, but actually whatever biome you have or any individual have is talk about the feeding behavior and the type of habitat the animal lives in. So if it is living in a good habitat with good food resources habitat, so it tend to have a balance or a normal gut microbiota. But if the person or the animal, if we talk about the Arunachal macaque, if it lives in a degraded, disturbed landscape where not food resources are there, or animal are depending on the anthropogenic food or the food waste that has been discarded by the human, so and they are eating those, so they got a dysbiosis of gut microbiota. So gut microbiota is also responsible and give you the signals to understand the physiological condition of the animal. For example, sometimes we eat something outside or junk food and we get the stomach upset. We get the diarrhea, sometime entamoeba, stylitic happen, or anything, we say food poisoning ho gaya, and then is a dysbiosis. So gut microbiota is important to understand the physiological condition, and is talk about the long-term association of the animal with a given habitat. So while we're working on the Arunachal macaque, we try to understand the gut microbiota. So is a kind of a different composition, is a coronagraph, and is talk about the bacterial community, Firmicutes, Clostridids, so different sort of bacterial communities present. When we compare this informations, the cumulative composition or the diversity of gut microbiota of Arunachal macaque, surprisingly it resembled with the human being. And this is again a question: how possible Arunachal macaque gut microbiota resembles with the human being? However, the rhesus macaque and Tibetan macaque, they also found in the in the same elevation zone, and their feeding behavior also almost same. Arunachal macaque and the Tibetan macaque, the feeding behavior is almost same. So we expect the gut microbiota should resemble with the Tibetan macaque, or might be the rhesus macaque, because they're largely frugivore, or eat on the wild berries and wild fruits. However, it is resembling with the human being. So then we have gone to the field and understand and watch their behavior. So they were good anthropogenic disturbances, and it is basically an example of anthropogenic provisioning. So they were eating on the garbage sites or dumping sites where this human are inhabited in inhabited nearby their habitats. This is again the phylogeography of the ibex, and is a very fantastic finding, and we have taken into account the all data that is available from all region in the country. So if you see here, this grayish part is the entire distribution of this species, the Siberian ibex. So what we all know that here in India, in the Trans-Himalayan region, this is a Trans-Himalayan species. I mean, found only in the Trans-Himalayan part of the country, like Ladakh and the Spiti region. So what the global communities knows that we have the Siberian ibex. So Siberian ibex is called the 𝗕𝗏𝗁 𝗁𝗏𝗁. This is the IUCN page. IUCN shows the distribution of Siberian ibex in India. However, when we did the genetic analysis, we found this is an alpine ibex. The pink color is the alpine ibex, which is found in the Europe, and this is a distribution of the Nubanian ibex or 𝗕𝗏𝗁 𝗔𝗏𝗁. So there are 3 species of ibex: 𝗕𝗏𝗁 𝗁𝗏𝗁𝗁, which is known by the global community. 𝗕𝗏𝗁 𝗁𝗏𝗁𝗁, this is the distribution of 𝗕𝗏𝗁 𝗁𝗁𝗁𝗁. This is the distribution of the 𝗕𝗏𝗁 ibex, also known as the alpine ibex. So alpine ibex is known to occur in the Europe region, and this is 𝗕𝗏𝗁 𝗔𝗏𝗁. This is a Nubania ibex, or is found in the Gulf part. But interestingly, when we did the genetic analysis, this is IT clade, where we have shown here this is IT clade. This IT clade is surprisingly clustered with the alpine ibex. So we do not have the Siberian ibex. We have the closest relative is the alpine ibex. So it's the eye-opening results, and we are now claiming this is a new species of ibex, and we are in the verge of this paper has been already published. So we are authenticated this population is not the Siberian ibex, but must be a new species which has to name it. We also work on pangolin, and some of you, if they are vigilant, they must be hearing some news that pangolin has good in trade. People are killing pangolin for their demand of the scales. They are scaly animals, and these scales has a reported use in the Chinese pharmacopoeia. Like we have a Ayurveda, and all the most plants are used for different purposes. There many animals are listed for their pharmacopoeia use. So since I'm doing the forensic analysis and forensic investigation, so I keep receiving cases of pangolin scale seizures. So we have done some morphological assignment, because the scales looks pretty similar. There are 2 different species. One is called Indian pangolin, which is Here is the blue color shows the distribution of Indian pangolin, and is a Chinese pangolin, little smaller in size and little darker color. It has a restricted distribution found only in the northeastern part of the country. So this yellow part is the distribution of the Chinese pangolin. But when you get this case like this, it would be difficult to assign whether Indian pangolin or Chinese pangolin. So I was working on a handy technique and tools where we can assign the two species separately, Indian and Chinese pangolin, and we have done some morphological measurements like OA, BC, their curvature, the weight, their quadrantal area, and this is a caudal scale. So again, this is a triangle in shape. So the curvature angle. So many criteria has been considered, and now we have been, we have established the signature of Indian and Chinese pangolin by just doing the small morphological analysis and measurements. So because there are certain regions where both the species, they overlap. So if any seizures come from any overlapping region, we can still assign them to a different species using the morphological analysis. We have also done the SCR analysis. Is like last year we have published a paper in 𝗕𝗶 pangolin indexing system, where now we have using DNA analysis we can assign is the Indian pangolin and is the Chinese pangolin. So we have generated a kit of 8 STRs where we can categorize Indian and Chinese pangolin separately. The Himalayan brown bear and the samples we got from the Himachal and the Uttarakhand is from Uttarakhand only, all samples. So this is how the scat or the fecal samples look like in case of Himalayan brown bear. So we have collected sample from Uttarakhand, and then with the 8 STRs, we have identified 6 unique individuals or genotypes, and we did not plotted them in a different scale. So again, the Asiatic black bear. So we have collected samples from Uttarakhand, Jammu and Kashmir, and 7 from West Bengal. They were also assigned to the different populations. So all analysis I just would like to highlight again is the scat of the animal, and our entire DNA efforts and work lies on the the samples we collected, because we cannot get the blood sample or tissue sample from the animal. So we try to use the non-invasively obtained their feces for genetic analysis. The kind of a demographic analysis we have done for different species is for the Himalayan or the Siberian ibex, red panda, Arunachal macaque, and the pangolin. This talk about the change in the population size over the time. So this 0 stand for the current time, and this is back in the years. So this is in million years. So 35 million years ago, the population was like this. So population has been stable in case of the Himalayan ibex or the Siberian ibex. However, if you see the red panda population, so this has recently declined. So demographically declined population in the recent years. So similar pattern we observe in case of the pangolin, Chinese pangolin. So if you see is the 5,000 years ago, the population was stable, but if it is approximately 2,500, 3,000 years ago, the population started declining. So it has declined in the population, and is a real-time estimate and prediction using the genetic analysis. And as for the Arunachal macaque, the population slightly declined, but now probably it is increasing or stable in. So this is another very good example where we have named a new species based on the phylogenetic analysis. So we collected one sample of fish, and it's a cold water fish. We were not able to assign them where it is. So what we have done, we have barcode this. We have generated the COI segment of this fish. So so when we have done the COI analysis of this fish, this is here how it categorized with the 𝗕𝗶𝗿 microbes. But again, this microbes was sequenced from the Tibetan region. So it's a Tibetan plateau region. So it was sequenced by some Chinese scientists. So the species we got in the 𝗕𝗏 river is a Chandrabhaga river which passes through this 𝗕𝗏 region. We are now assigning this to a new species. We are naming this as a new species, which is closely related with the 𝗕𝗶𝗿 microbes, but it is far, genetically differ, 2 to 3% different from this species based on the COI, and this species which is reported from the earlier from the same region is about 12% different. So we are saying the species we got from 𝗕𝗏 region, it is genetically very different from the other possible species, genetically possible species reported from the same region. So we are now trying to name it. This paper is under consideration. 𝗕𝗏𝗰𝗲 is into the business of training and capacity building. We keep organizing training, workshop, awareness program for the students and the law enforcement, and for the forest department, 𝗕𝗏𝗰𝗲, Wildlife Crime Control Bureau, and other academician and non-academician institutes. So we keep organizing these workshops. Now, as a curiosity, being a biologist and biotechnologist, we have done some analysis in the COVID. So my talk is completed. I mean, what I propose is complete up to this slide, but here I'm just discussing something new. And because everybody was talking on the coronavirus, and we were in the lockdown since March and up to now, very recently it's all open. So during the lockdown, I started this project. It's like a home-based assignment we have taken with our curiosity and the skills we have, the biotechnological skills we have. So just bear with me. I would take another 10 minutes. I hope it is okay with you, although I'm very fast, but I hope I'm still meeting the time. Do I have a 15 minutes more? Hello?
Yes, sir. Yes, sir.
Yes, sir.
Go ahead.
Can I take 10 or 15 minutes more?
Yes, sir. Sure, sir.
Okay, so this we have taken as a curiosity. So we wanted to understand the spread and transmission of coronavirus in the country. Okay, so this is a very current estimate. Over 90 million people has been infected by coronavirus, and about 2 million deaths in reported in 216 countries. I mean, everybody know is a pandemic, and worldwide the entire globe has been affected. I'm not discussing all this. This all data is known to all of us. So this is how the growth has initiated decline. I'm not discussing, don't have time for this, but something which anyone can do. I mean, you studying biotechnology or bioinformatics should understand, and anyone, I mean, we don't need any type of funding for this, or we have done all this in silico analysis of this. And if you see by my name, we have also developed a corona vaccine in silico approach. The paper was published in Nature in scientific report, and this is a half part of the study that we have done. This study I'm going to discuss also published in PLOS ONE 2 months ago. So this is a kind of a coronavirus genome. It is about 30,000 base pair in length, and it has a 4 structural gene: S gene, E gene, M, and N gene. So S stand for the spike gene or the surface glycoprotein, what we know. E stand for the envelope protein, membrane protein, and nucleocapsid protein. These are the 4 structural protein, and other 8 are the non-structural protein, where ORF1ab is the largest part of imparting the largest part in their genome. It is a single-stranded genome of the virus. So people have a confusion whether it is a man-made virus or it has spread through bat using, I mean, through pangolin as an intermediate host. So let me discuss briefly one paper that published in, I think it was published in Nature itself. So is the genome of the coronavirus the spike gene, and if you see the spike protein here, spike amino acid sequence here, so this part has then again subdivided, I mean, spelled out nicely. So you see here there are certain complementary site that matches with the bat also, and here some matches with the pangolin also. So these are the nucleotides which are in blocks, right? It matches identical with the pangolin and the bat. Here also some amino acid, they matches with the pangolin and bat. So this is the first paper that talk about that coronavirus is not man-made virus, but it is a naturally occurring virus. I am not supporting or doubting this study. I'm just briefing you that what has been published, and this paper came very early. I think it was published in last year. January 2019, 20. So this talk about this because it has some complementary site with the pangolin and the bat coronaviruses. So that is similar, and it might have originated through bat from a pangolin as an intermediate host. So again, how different population of the globe, they respond differently to the coronavirus. People were talking like we have been raised in a dirty atmosphere, or we are more resistant to the virus because we had a BCG vaccine during our childhood, or those countries, developed countries which are more clean, tidy, they are more susceptible. We have a little resistance because we have been keep exposing with the different viruses during our childhood. So there are many sort of things were there. So one study that published in Cell Discovery is again a cell research journal that talk about the variants in the ACE2 gene. So ACE2 is the one receptor, is a human anti-angiotensin converting enzyme. ACE2 is a receptor which actually perceive the virus. Do I have time? Anyways, I'm getting some message. I'm ignoring this. Please let me know when I have to close this. I would be going faster. So ACE2 gene, one study has mapped the different variants of the ACE2 gene all across the globe. And this is the SARS is the Southeast Asian human population. So if you see the frequency of variants, they vary between the human population, the human population data. So ACE2 receptor, which is the first molecule that perceive virus, and then virus get the passage to enter into the human body, right? So there is a frequency variation between the different alleles. So in these South Asian countries, there is a difference in the allele frequency. Different 9 alleles has been reported, I think so, and there's a different variability. So America has a different variability. So all this, if there is a red, that means there's a high frequency of these alleles, because the occurrence of those different receptor vary among human populations. That is why we have a different response to the virus. What we have done, we have taken the coronavirus genome that has appeared into the India in a very synchronized or I should say time-lapse manner. So we have since the very first corona case has been reported on 30th Jan to 3rd Feb, we have been keep organizing and analyzing data and try to understand how it has been spread. So we have this chronological order how the cases has appeared or observed in the country. Feb 21st, that was a 23 Italian tourists that was found infected with corona. They were before they get infected, they hired actually a cab in Delhi and then traveled to Rajasthan. The driver was also infected. I think this all you must be knowing. So anyways, we have registered all these cases and incidences, and along with we have also arranged the data in different time scales. So up to, we say T1, that all 35 SARS-CoV genome up to 21st April. So we have the data on public domain of 35 coronaviruses up to 21st April. The T2 time to T2 says 53 coronavirus genome we have up to May 2, and T3 up to 112 SARS-CoV-2 have up to May 5. So this is what the data that was available in the public domain on NCBI and other sources. So we have did the phylogenetic analysis. So we have here categorized. These are the first 2 cases that appear in Kerala, right? This is they cluster along with the Wuhan HU1 genome. So that is for sure that India, the first case that was reported from the Wuhan only, and we know the travel history of the patient also. The girl was studying in Wuhan Medical College, and she traveled to from Wuhan to Guangzhou, and Guangzhou to West Bengal, in Kolkata, and then Kolkata to Kerala. So that first case was really clustered along with the Wuhan 1 genome. And then the second case, this is then a chronological order. So we have taken these all samples clustered with the Iran genome. So and these all 3 the Italian clusters. So we say in India, the initial visitors from Italy, Iran, and China, they brought coronavirus in the country as per the chronological order of their appearance. in the public domain. This is the same depicted in the haplotype network. In the this is again a comparisons with the T1 time, T2 time, and T3 time, how the coronavirus has started spreading. So up to here we have just ancestry from 3 different countries, but here then starting proliferating and starting having the community spread. Up to T3, if you see, is a different state name also given: Delhi, Tamil Nadu, Odisha, Haryana, Maharashtra. So it's become a star-like phylogeny here. So this become very prominent. This spread widely in the country. This haplotype, become very frequent, prevalent, highly dominant, and spread it out the country. So we try to understand. So you say the May 5th we have a community spread, because these guys were not having any travel history, but they still had the same haplotype like 𝗕40. So the 𝗕40, which was the primary traveler or having a history of travel abroad, the same like here also. So we had the signals of the community spread of the virus up to the May 5, although ICMR has declared very late. And then we have keep adding on the genome in this, and we keep analyzing it further. So we have analyzed about 400 genomes in here, and in every state we got this kind of bar plot pie chart. So many variants in different countries. If you see here, the Gujarat, the Delhi, and Mumbai, deeply affected and with having several variants. So we got 198 variants up to May 5th. And then we did the demographic analysis also. We tried to understand how long the coronavirus will remain, increasing. So we have a very 3 months data. So keep studying the demography. The coronavirus population size was increasing or fluctuating up to the 26th April, and this actually coincides with the real cases that we observed. We also tried to understand the different mutants we got, because we were thinking like whether is there any mutant specific transmission, is there any specific mutation which is more virulent or pathogenic in nature. So we try to prioritize those mutations that happen in the spike gene, and that too in the RBD. RBD is the receptor binding domain. If you remember my first slide where I was discussing about the ACE2 receptor, right? So ACE2 receptor and RBD, these two bind together, and that virus get the passage to enter in the human body. So we prioritize all those mutations that happen in the RBD side. So we got there are a couple of mutations which is in the RBD, and there was one mutation that we found in approximately all the genome that we analyzed. So this mutation D614G was really something fuzzy and little naughty also, I would say, because coronavirus, what we were knowing, it is getting mutated in every 15 to 20 days. So they are accumulating one mutation in 15 to 20 days. And this RBD side also, we tried to understand these mutations. This mutation was everywhere, D614G, the most prominent and most abundant mutation in the coronavirus, up to the 400 genome that we have analyzed. And then this is how we have also tried to understand how their interaction happening with the ACE2 receptor. And just would like to highlight here D614G. This mutation, I have termed this the M4 mutations. So M4 mutations here, M4 and M5 mutation having a high binding affinity with the ACE2 receptor. So what we are suspecting that this particular mutations is actually increasing the binding affinity. So just two molecules, if they are trying to attach together, and this mutations occurred, so it increased the binding affinity, and that is how this mutation was prominent. We try to also understand their vibrational and interaction with pattern with the different mutants. So this is somehow, I mean, we have also calculated how it is affecting the the binding affinity of this. So with this, this paper was also published in PLOS ONE. I have many more things to discuss, but I have just limited my talk. I wanted to discuss with the vaccine concept also, which we have done by ourselves. It is a in silico designing of the vaccine, and the paper was published in scientific report in Nature journal. So yeah, I would like to just stop here, though I have so many things to present. Yeah, I would like to thank all the faculty members of the college, and particularly because I had interactions and 2-3 times discussion with Dr. 𝗕𝗶𝗁𝗏𝗏𝗏𝗏. So I would like to thank him again, choose me, giving me this opportunity And again, would like to acknowledge all the other faculty members, and also all the organizer, the team, because it's a team effort, and this whole day we all have been busy here. And so I would like to thank one and all. Yeah, thank you so much.
Thank you, sir. Thank you very much, sir, for giving us an informative and an illuminating lecture. And we also thank you for sharing your recent research works with us.
If anyone has any questions, any curiosity, I'm here to answer you. And if not, and if we still have some time, then I can show you a small video just for recreation or getting you charged again.
Our students are really very interested, sir, and we have a lot of questions, but due to the lack of time, we are ending the session and we will forward it to you.
Sir, just we can have a video, sir. Yes, sir. Hello.
Yes, yes.
Yes, sir. We have an interesting thing. We can show that video.
Yeah, I hope that the student would be very
yes, of course.
The field effort. So I would like to just share with them that it's not like and
yes,
I have two videos, one made by the student and one made by the professionals. I would like to show the video made by the students so that they'll get more attachment. Professional is such a super classy video, but I would like to show you the one that has been made by the student with their mobile cameras only.
I hope this will be better. Any queries, I will give the email. Your email, they will have a contact personally. Okay, because of lack of time, it's better to have videos. Yes, you can proceed.
Yeah, I'm just sharing my. Is it visible?
Yes, sir. Yes.
Audio is visible?
Yes, everything is okay.
And that has been made by the students only. They work in Eastern Himalayas, and there they have shooted with their own mobile cameras. So is representing the field activities, how difficult the field is, and they climb Himalayas and in search of the field samples. This is how the fecal droppings looks like in field. So they are interviewing locals.
Okay. Crossing the river.
The leech.
Oh,
all are CSIR JRF, and they are studying different species for their PhD, setting up the cameras for taking the photographs of animal. So it's a very harsh and adverse condition where they do the science surveys, and they actually, stay in the camps and the forest rest house. They cook by themselves. They also carry the rations. all the field gears they carry on their backs. So he's in Spiti, is the Western Himalayas, in the Spiti region. And here we have put several camera traps for recording snow leopards in brown bear. So they have a GPS also. They record the animal presence, and this is how sometimes they have to cross rivers. So if you have to go from one side to other, it's a challenge for them. Yes.
Thank you very much. Really fantastic information you showed us within few minutes, and also we can able to assess the data what you presented. It's not lightly collected, how much hardship is behind the data. So thank you very much, and also special thanks on behalf of that the interest is shown on our students, particularly to show these videographs. Thank you very much. Thank you,
sir.
Over to Biotechnology program.
We can have some questions if you want. Otherwise, my email.
Okay, so just as I mentioned, I will give the email. Okay, they will contact because lack of time, many has to rush back here. Okay, it is rainy here, sir.
Okay.
Okay, thank you.
One and all gathered here. I heartily welcome you all to the validatory session of Second International Conference on Recent Trends in Biotechnology 2021, Jamal Muhammad College. We had deep insights into several recent trends in biotechnology, and we have had productive and inspiring time together. A day without prayer is a day without blessing, and a life without prayer is a life without power. Now I call upon 𝗕𝗶𝗿𝗄𝗏 of 3rd BSc Biotechnology, Jamal Muhammad College, to recite a few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ألم نشرح لك صدرك ووضعنا عنك وزرك الذي ينقض ظهرك ورفعنا لك ذكرك فإن مع العسر يسرا إن مع العسر يسرا فإذا فرغت فانصب وإلى ربك فارغب.
Thank you, Rabia. Now, I kindly call upon Dr. 𝗕𝗶𝗵𝗏 sir, MID and Assistant Professor of our department, to deliver the welcome address.
Good evening, one and all gathered here. It is my great pleasure to welcome
It is my great pleasure to welcome our most honorable secretary and correspondent, honorable treasurer, dynamic assistant secretary, most respected principal, vice principal, additional vice principal, young and energetic directors, former heads of our biotechnology department. I wholeheartedly welcome you all to the valedictory function of Second International Conference on Recent Trends in Biotechnology. I am very much happy to welcome our honorable chief guest, Dr. 𝗕𝗶𝗁𝗏𝗏𝗏𝗏, Scientist C, Center for DNA Taxonomy, Molecular Systematics Division, Zoological Survey of India, Kolkata, West Bengal, India. It is my great pleasure to welcome our Biotechnology Department coordinator and faculty members. It is my honor to welcome the neighboring Department of Microbiology coordinator, member in charge, and faculty members. I also welcome our Biotechnology Department research scholars and students. I am happy to welcome heads, staff members, and students of Zoology, Botany, and Sisters Department on this occasion. I welcome the participants from various colleges and universities in and around the globe of today's program. Once again, I welcome you all. Thank you.
Thank you, sir, for your sweet words of welcome. The science of today is the technology of tomorrow. Now I invite Dr. 𝗕𝗓𝗏𝗏𝗏𝗏, sir, Scientist C, Center for DNA Taxonomy, Molecular Systematic Division, Zoological Survey of India, Kolkata, to address the audience again, to deliver the valedictory address.
Yeah, thank you, madam. And it is a great opportunity for me to interact you again. And I would say that the entire session since morning I was attending all the two lectures that early happened. The speakers were really great, and the organizer has meticulously identified the speakers. The first speaker, that was also the alumnus of your college, Dr. 𝗕𝗶𝗁𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 really a great speaker, and also has done a marvelous job. And it was really very interesting to listen the mouse metagenetics projects. And the second speaker, that the scientist from the ITRC, Dr. 𝗓𝗂𝗓𝗂𝗓𝗂𝗓𝗂𝗓𝗓, he talked about the microbial diversity and their different role in bioremediation. It's a classical job. He has presented several examples, and definitely, I mean, this sort of work is actually required, because in country we have a huge problem to deal with the polluted sites. So in particular, the bioremediation of the polluted watersheds and the soil. So he has really shown a very good example, and the possibilities that one has to invest more in this field to tackle these adverse situation. At the same time, I also feel like we should also create an awareness among the communities, and I mean it should come in within to stop these activities. And I hope that my presentation would be also fruitful in terms of giving you a deep insight of the wildlife conservation and the use of molecular biology tools. And all the paper presented by the students was very good. And I would say a couple of them, they were more concentrated on the antimicrobial or antibacterial activities. So I was expecting little more diversity in the chosen topic. So I can see, I think 3 were on a little different one, like the ligament tissue engineering and the analysis of the Pfizer vaccine. But I think there could be more diversity in the presentation. So otherwise, most of them were dealing with the some or other plant extract and dealing with the antimicrobial. I'm not saying that they were not significant. but when you have a bigger audience, and so I expect diversity of the topic should be there. So all the presentations was really good, and your faculty, all the students, I would like to highlight that you are really so fortunate that you have such a good academic faculties who think about all these kind of events, and this is indeed required at your age, because once you are exposed to this kind of activity, so you get more different exposure. And there could be some poster talk, if there could be e-posters could be accommodated, instead of having the presentation, if can have a e-posters, just in one slide one has to discuss the key findings. I would like to thank all the faculty members, the team of organizers, and this is on and off the record in the background, because it's you need to have a good team efforts to organize such a big event where you have included approximately more than 70 students. I hope there were more than now also 79 participants online. So it was, to me, it was really a good experience. And I'm always a kind of a open-minded scientist. I always like participating and presenting work in different forum, and is a very good forum where I can interact with the young students. I can enlighten them that these are the different dimensions of the application part of the biotechnological tools. So it was really nice, and I congratulate all the young scientists there coming up, and budding scientists, I would say rather. And I thank one and all of the PG and Research Department of Biotechnology. And yeah, thank you so much. It was really a wonderful experience for me to join you here. Yes, this is what
Thank you very much, sir, for your enthusiastic and encouraging words.
Now it's time for the announcement of prize winners of today's presentation. I kindly invite Dr. 𝗓𝗂𝗍𝗓 sir, the coordinator of our department, to announce the best paper presentation of this conference.
Thank you very much. Apart from this, I take this opportunity to thank Dr. 𝗓𝗂𝗍𝗓 sir for your active participation. Even though we thought that we'll be in the evening hours, we are very much happy that you are there and you are viewing our work. And thank you very much for your compliments, sir. So, as you said, nearly we had more than 250 participants. Actually, only 100 can be accommodated in the Zoom. The remaining participants are in the YouTube. So that's for information. So it's not only 70 are here. So in the YouTube, there are more than 100 are viewing our program. So your lecture has reached more than 200 now. So you can get that satisfaction. And definitely this one, that is because of your lecture, definitely few will take this job even in the field of conservation biology. So with this information, I will proceed with the job assigned to me. So here nearly about 25 papers were received. So only 14 can be accommodated because of lack of time. So among these, I am happy to announce some best presentation. It's not best presentation, based on the performance we are here, all 14s and almost everyone did their best. But within this, we have to categorize because of our warrant of proceedings. Here, the first paper, best paper presenters, and first prize goes to 𝗕𝗶𝗁𝗂𝗍𝗓 Manoormiyam Sundarnagar University, Tirunelveli. First paper, and paper second best paper that is goes to Sudha, 𝗓𝗏𝗏 Andor Arts and Science College, Tiruchirappalli. So third prize goes to Sri Anita, that is Malankara Catholic College, Kanyakumari. So this is about the three best papers we have chosen. So congratulations to the best paper winners. Then over to MCQs. Thank you very much.
Thank you, sir. We congratulate the winners. The second International Conference on Recent Trends in Biotechnology 2021 was made possible by many hands, hearts, and minds working together. To express a word of appreciation and gratitude, I kindly call upon Mrs. 𝗕𝗶𝗁𝗲𝗵𝗲 ma'am, Assistant Professor of our department and the Organizing Secretary of the conference, to present the conference report and deliver the vote of thanks.
It's my privilege to propose that I vote to compile for the one-day international conference on recent
இந்த ஹெட்ஃபோன் யூஸ் பண்ணிக்கோங்க. Good evening to one and all gathered here. It's my privilege to propose the report to compile for the one-day international conference on recent trends in biotechnology. The session begins with a welcome address delivered by Dr. 𝗕𝗶𝗁𝗲𝗵𝗲, convener, Department of Biotechnology. Our Honorable Chief Guest, Dr. 𝗕𝗶𝗁𝗲𝗵𝗲𝗵𝗲, delivered the inaugural address, and Dr. 𝗕𝗶𝗁𝗲𝗵𝗲, Assistant Professor, Department of Biotechnology, proposed the vote of thanks. The first session begins with the invited lecture 1, delivered by Dr. 𝗕𝗶𝗁𝗲𝗵𝗲, EMBO, Hilbom Research Fellow, Department of Biotechnology and Cellular Physiology, Stanford University, USA, on the topic Mouse Megagenetics Gene Function at Large Scale. This was followed by the second invited lecture on Exploring Microbial Diversity and Metagenomics for Bioremediation of Persistent Pollutants by Dr. 𝗓𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏
T or Lucknow. The second session began with paper presentation of with Dr. 𝗕𝗶𝗿𝗄𝗏 and Dr. 𝗓𝗏𝗫𝗏 as the moderators. Overwhelming responses received from the student participants. Among the 25 research articles received, 14 papers selected for paper presentation. This was followed by interesting lecture, Role of Conservation Genetics in Wildlife Conservation by Dr. 𝗕𝗏𝗁𝗏, Scientist C, Center for DNA Taxonomy, Zoological Survey of India, Kolkata. The welcome address for the valedictory ceremony was delivered by Dr. 𝗕𝗶𝗁𝗏, Assistant Professor and Member in Charge, Department of Biotechnology. The valedictory address was delivered by Dr. 𝗕𝗏𝗁𝗏. Thanksgiving is our tradition and customs. It's my great honor to propose the vote of thanks on this occasion. First of all, I would like to thank the Almighty for making this program our grand success. I thank our esteemed secretary and correspondent, our respected treasurer sir, and our assistant secretary, member and honorable director for their motivation and guidance. Thank you, sirs. A special thanks to our beloved principal, vice principal, sir, and additional vice principal. Thank you, sirs. I express my heartfelt thanks to the directors of hostel administration. Thank you, sirs. I would like to acknowledge my gratitude to our honorable invited speakers, Dr. 𝗕𝗏𝗁𝗏, Dr. Nadeshan Manikam, and Dr. 𝗕𝗏𝗁𝗏, and thank them for their exemplary lectures. Your talk are a great encouragement to all of us and to the budding student scientists of our college and various other esteemed institutions. Thank you, sirs. My deep sense of gratitude to our coordinator, Dr. 𝗕𝗶𝗁𝗏𝗏, for his support and encouragement. Thank you, sir. I shower my heartfelt thanks to the member in charge and all my colleagues for their timely assistance and valuable contribution, and thank the non-teaching staffs for their timely help. Thank you all. To conclude, my heartfelt thanks to faculties from various other colleges, institutions, and universities for their active participation. Last but least, thank you students for your attentive presentation. Expect the same in all our future endeavors. Thank you, everyone.
Thank you, ma'am. We have an announcement. We kindly request everyone to fill up the feedback form, which will be provided in the chat box now. You will need to fill the feedback form in order to receive the e-certificate. So kindly please fill the feedback form. Thank you. I repeat, we kindly request everyone to fill up the feedback form provided in the chat box. You will need to fill the feedback form in order to receive each certificates. So please kindly fill the feedback forms. Thank you.
I finally thank everyone for joining us and made this event a grand success. I conclude by saying that let us all be guided by the things we have learned and heard throughout the conference and be able to see and influence our future. Hope we meet up soon in the next upcoming conference. Thank you all. Have a nice day.
Just home screen or